January 23rd, 1960. Two men are descending into the Mariana Trench inside a steel sphere, barely large enough to stand in. They have been falling for nearly 5 hours. The pressure outside has reached 16,000 pounds per square inch. At some point during the descent, the outer viewport cracks, not shatters, cracks. They keep going. When they finally touch the bottom, nearly 11 kilometers below the surface, they switch on the exterior lights and something is looking back at them. A flatfish, about a foot long, white, both eyes on one side of its head, sitting on the seafloor at the deepest point on Earth, as though nothing about this environment is unusual. Jack Picard later admitted they were astonished to find complex vertebrate life down there at all. But that fish was not lost. Something like it had been living in that darkness for millions of years before humans ever arrived, perfectly adapted to conditions that should make vertebrate life impossible. And it was far from the strangest thing they never knew existed. There are anglerfish whose males fuse permanently to the female's body, dissolving into her bloodstream until only their reproductive organs remain. There are fish so black they absorb almost every photon of light that touches them, disappearing into the darkness more completely than some modern stealth materials. There are giant single-celled organisms, larger than dinner plates, building intricate structures from glass. There are worms over 8 feet long with no mouth, no stomach, and no digestive system, and there are entire ecosystems that have never depended on sunlight at all. None of this is an accident. Every one of these creatures was shaped by the same environment, an environment defined by crushing pressure, permanent darkness, near freezing temperatures, and a constant shortage of food. In this story, we're exploring how life adapted when almost every condition it depended on was taken away, long before any creature learned to glow, fuse, vanish, or endure. The deep ocean had already rewritten the rules. It did so by taking away 4 things that life on the surface treats as ordinary, not one at a time, but all at once, and for spans of time so vast that entire branches of evolution rose and disappeared while the conditions remained unchanged. The first removal was pressure's opposite, freedom from weight. At the bottom of the Mariana Trench, water presses downward with a force exceeding 1,000 atmospheres. More than 15,000 pounds bear against every square inch. Yet the danger is not simply that bodies might be crushed. The deeper consequence unfolds within living chemistry itself. Every cell depends upon proteins folded into precise shapes. These microscopic structures act as workers, messengers, builders, and regulators. Their form is inseparable from their function. Under immense pressure, those forms begin to distort. Enzymes lose efficiency. Cellular membranes become increasingly rigid. The processes that sustain life start to falter. By roughly 400 to 500 atmospheres, many organisms can no longer maintain the chemistry required for survival. The hadal depths exist far beyond that threshold. Then comes cold. The water resting in the deepest trenches remains between 1 degree Celsius or 34 degrees Fahrenheit and 2.5 degrees Celsius or 37 degrees Fahrenheit. In such conditions, biological activity slows dramatically. Reactions that occur almost instantly within warmer bodies proceed with extraordinary patience. Molecules drift, collide, and transform at a pace that would seem unfamiliar to organisms nearer the surface. Metabolism approaches a state that resembles suspension without ever fully arriving there. Life continues, but it does so with remarkable restraint, conserving every possible fragment of energy. Darkness follows, not the darkness of evening, nor the darkness behind closed eyes. The floor of the Mariana Trench has never received sunlight. Across millions upon millions of years, no dawn has reached it, no season has altered it, no passing cloud has mattered. Light is not missing there. Light is simply absent from the design of the environment. The condition extends so far into the past that it predates humanity, our ancestors, and countless forms of life that once dominated the planet above. And then there is scarcity. From the surface, a slow descent begins. Tiny fragments of life drift downward through the water column. Dead plankton, broken tissues, fecal particles, fading remnants of organisms that completed their lives far above. Collectively, scientists call this marine snow. The name sounds gentle, but by the time this material reaches the seafloor, little remains of its original richness. What arrives is sparse, unpredictable, and often nutritionally poor. The abyss receives food the way a desert receives rain, irregularly and in quantities that rarely satisfy. Any one of these conditions would present a formidable challenge. Pressure exists elsewhere, cold exists elsewhere, darkness exists elsewhere, scarcity exists elsewhere. Yet the deep ocean is shaped by their simultaneous presence. They overlap continuously, reinforcing one another through hundreds of millions of years. No pause interrupts them, no refuge escapes them. Every organism that persists there must negotiate all four realities together. This combination becomes an engine of selection, refining life with relentless consistency. The strange anatomies, unusual behaviors, and improbable survival strategies found in the deepest water are not separate mysteries. They are products of the same underlying machinery. Before any creature can solve the problems of food, reproduction, or perception, it must first solve the problem of pressure. Without that solution, nothing else can endure long enough to matter. And in the immense darkness beneath the world, life eventually discovered a remarkably elegant answer. Deep within the tissues of hadal fish, carried through blood and packed between cells, drifts a molecule so small that it escaped notice for much of scientific history. It has no striking shape, no dramatic appearance, no obvious sign of its importance. Yet without it, the deepest vertebrate life on Earth would simply cease to function. The molecule is called trimethylamine oxide, and in the deepest trenches, it performs a task upon which entire lineages depend. Near the surface, trimethylamine oxide exists in modest amounts. There, it is little more than a routine participant in metabolism, one chemical among countless others circulating through living bodies. But as fish descended through evolutionary time into darker and deeper water, this ordinary compound acquired a new purpose. Pressure was constantly pushing against the machinery of life, distorting delicate structures that cells relied upon. Trimethylamine oxide became a kind of molecular guardian, surrounding proteins and helping them retain the precise forms required for their work. The relationship between depth and trimethylamine oxide is remarkably consistent. Species living farther below the waves carry greater concentrations within their tissues. In many cases, measuring the amount present in a fish reveals roughly how deep that fish spends its life. The descent into darkness is recorded directly in chemistry, as though every meter traveled downward leaves its own signature behind. This pattern reaches one of its most refined expressions in the Mariana snailfish. Hidden within its genetic inheritance are multiple copies of the instructions responsible for producing the enzyme that generates trimethylamine oxide. Researchers discovered 5 copies of this critical genetic sequence. Over immense spans of time, evolution appears to have repeated the instructions again and again, preserving a process too important to risk losing. The result is a body saturated with the chemistry needed to endure pressures that would dismantle the biology of most vertebrates. For many years, Scientists believed this adaptation might also reveal a final boundary. As depth increases, so does the concentration of trimethylamine oxide required to stabilize proteins. Eventually, a limit appears. Fish tissue can hold only so much. Beyond a certain point, additional molecules can no longer be packed into the body. Calculations suggested that this ceiling might occur at approximately 8,200 meters, which is about 5,096 miles. At that depth, trimethylamine oxide would approach physical saturation. The chemistry supporting vertebrate life would reach its maximum capacity. The idea carried an elegant simplicity. Somewhere in the deep ocean, there might exist an invisible line, drawn not by geography or geology, but by molecular physics. A fish could approach it. A fish could perhaps live upon it. Beyond it, vertebrate life might become impossible. Then the ocean offered another reminder of how incomplete every map remains. In 2023, researchers recorded a snailfish at a depth of 8,336 meters, which is about 5,180 miles. The observation exceeded the proposed boundary. The line did not vanish, but it shifted. The limit moved farther downward into the dark, and scientists adjusted their understanding once more. The chemistry remained valid. The ocean had simply revealed that the calculation was not yet finished. Even when brought upward, trimethylamine oxide continues to tell part of its story. Deep-sea fish often release a powerful ammonia-like odor after reaching the surface. The smell comes from the very compound that helps sustain them below. Removed from extreme pressure, the chemistry begins to break apart and transform. A molecule that protected life in the trenches becomes unstable in the comparatively gentle conditions above. There is something striking in that reversal. The same adaptation that allows survival in one world becomes a disadvantage in another. The deepest fish are not built for every ocean. They are built for their ocean, tuned with extraordinary precision to conditions that exist nowhere else. Within their tissues, trimethylamine oxide preserves the architecture of life against immense pressure, allowing cells, proteins, and entire bodies to remain coherent where they otherwise could not. And once pressure yielded its answer, another challenge emerged from the darkness, shaping bodies in a completely different way. Imagine building a predator for a world where opportunity arrives only occasionally. Not every day, not every season, but at intervals so uncertain that missing a single meal might mean waiting through weeks or months before another appears. In such a place, efficiency begins to shape anatomy with unusual precision. Structures that help secure food expand, structures that do not become secondary. Over immense spans of evolutionary time, this process produced the female anglerfish. At first glance, much of her body appears organized around a single task. The head dominates her form. The jaws are immense relative to her size, opening far wider than would seem reasonable in shallower waters. Along those jaws sit rows of teeth angled inward toward the throat. Their orientation matters. Once prey slips between them, every struggle pushes it deeper. Escape becomes increasingly difficult because the geometry of the mouth itself works against retreat. The stomach follows the same logic. When empty, it remains compact, conserving space and energy. When food finally arrives, it transforms. Flexible tissues stretch outward, expanding into a translucent chamber capable of holding prey larger than the anglerfish herself. In some documented cases, fish have been swallowed whole, despite exceeding the predator's own body mass. Such meals appear improbable until viewed through the lens of scarcity. The deep ocean rarely rewards moderation. When nourishment appears, the safest strategy is often to accept everything available. Above her head, rises one of the most recognizable structures in marine biology. Extending from a modified dorsal spine is a specialized organ known as the esca. It hangs before her like a lantern suspended in open water. Yet the glow does not originate from the anglerfish alone. Within the tissue of the esca live colonies of bioluminescent bacteria, microscopic partners whose chemistry produces light. Different species possess different versions of this structure. Some are elongated, others branch into delicate projections. The brightness, shape, and arrangement vary from lineage to lineage, creating distinctive signals within a realm where visual communication is otherwise difficult. The esca functions simultaneously as lure, identifier, and beacon, all condensed into a single, elegant adaptation. The relationship between anglerfish and bacteria extends far beyond simple coexistence. Over generations, the bacterial inhabitants have shed much of the genetic material once necessary for independent life. Nearly half of their original genetic instructions have disappeared. Protected within the host, supplied with nutrients and stability, they no longer require many of the biological tools carried by their free-living relatives. The partnership has become so intimate that separation may no longer be possible. Host and microbe operate as parts of one integrated system, each shaping the survival of the other. Around this luminous structure lies another remarkable adaptation. The skin of many female anglerfish absorbs extraordinary amounts of incoming light. Rather than reflecting illumination outward, it captures it. The result is a body that remains almost invisible, while the glowing tip of the esca remains conspicuous. From a distance, prey encounters what appears to be a solitary point of light suspended in darkness. The predator behind it nearly disappears from view. This arrangement transforms the anglerfish into something unusual. She does not chase food across great distances. She does not patrol vast territories. Instead, she allows the environment to deliver opportunities to her. Energy is conserved, motion is minimized. The lure performs the invitation, and the jaws complete the transaction. Weeks may pass without a meal, sometimes months. Her metabolism is tuned to these intervals, slowing the expenditure of precious resources and allowing survival through long periods of absence. Every aspect of her body reflects this rhythm. The mouth, the stomach, the luminous lure, the light-absorbing skin all converge upon the same objective. The result is not a biological mistake or an evolutionary excess. It is an organism refined around a single environmental demand with extraordinary thoroughness. In almost every meaningful sense, the female anglerfish is a living trap, sustained by patience, shaped by scarcity, and carrying within her design one of the most specialized predatory architectures ever produced by vertebrate life. Yet even this remarkable creature is only part of the story of anglerfish. For decades, museum collections told an incomplete story. Preserved specimens arrived from deep water carrying luminous lures, oversized jaws, and all the distinctive features that made anglerfish famous. Nearly every individual examined was female. Researchers described species, compared anatomy, and built classifications. Yet one piece remained strangely absent. The males seemed almost to vanish from observation, existing more as expectation than certainty. When they were finally found, the surprise was immediate. Nothing about them suggested they belonged beside the imposing females already known to science. They were tiny, often measuring only a few centimeters in length. Their bodies lacked the elaborate structures associated with their partners. There was no glowing lure, no enlarged feeding apparatus, no indication of a life built around active predation. Compared with the female, The male appeared reduced, as though evolution had taken the same blueprint and erased most of it before completion. Yet, this apparent simplicity concealed an extraordinary specialization. The male enters life carrying a singular objective. Time begins working against him almost immediately. His digestive system is underdeveloped, providing only a temporary path to survival. Energy reserves are limited. The future narrows toward a single requirement. Somewhere in the vastness of deep water, a female must be found before those reserves are exhausted. This challenge is difficult to overstate. The search unfolds across immense three-dimensional space, through waters lacking familiar points of reference. Distances between individuals may be enormous. Encounters are rare. In such circumstances, many sensory systems become secondary. One remains essential. The male possesses an exceptionally refined sense of smell. Chemical signals released by females drift through the water, forming faint trails that can be detected even across great distances. His body is built around interpreting these signals. While other structures remain modest, the machinery devoted to locating a mate becomes extraordinarily sensitive. Every moment of his existence converges toward following those invisible traces. When the search succeeds, an unusual transformation begins. The male bites into the female's skin and remains attached. At first, the connection appears temporary, little more than physical contact. Then tissues begin to merge. Blood vessels link together. Cellular boundaries that ordinarily separate one vertebrate from another start to disappear. Under normal circumstances, this process would fail almost immediately. Vertebrate immune systems are designed to recognize foreign tissue and destroy it. They maintain the distinction between self and non-self, protecting the integrity of the body. In anglerfish, something remarkable occurs. The male's immune defenses effectively stand down. The biological barrier that would normally reject the female's tissue ceases to operate in its usual way. As fusion progresses, the male undergoes profound reduction. His eyes begin to degenerate. Internal organs lose their original function. Structures once necessary for independent survival are gradually absorbed. Even his circulatory system merges into hers, transforming two separate blood supplies into a shared network. Eventually, little remains of the original animal as a distinct individual. Attached permanently to the female's body is a small mass of living tissue supplied directly through her bloodstream. Within that tissue, reproductive function persists. Everything unrelated to reproduction has been relinquished. Researchers have collected females carrying multiple attached males simultaneously. In one documented case, 8 males remained connected to a single female. Each represented a completed search, a successful encounter converted into permanence. This arrangement is known as sexual parasitism, and among vertebrates, it stands alone in its degree of integration. Yet viewed through the lens of the deep ocean, its logic becomes strikingly clear. Populations are sparse. Individuals are widely separated. Opportunities to meet may occur only once in a lifetime. Under such conditions, temporary pairing becomes an unreliable strategy. The solution is not to improve the search indefinitely. The solution is to eliminate the need for a second search altogether. The first encounter becomes the last encounter required. The partnership is fixed in place. Resources are no longer spent locating mates. Biological structures no longer necessary are gradually surrendered. What remains is the minimum arrangement needed to ensure reproduction whenever conditions allow. The male anglerfish is often described as bizarre, but the deeper pattern is one of relentless efficiency. The ocean did not shape him into something incomplete. It removed every feature that no longer served a purpose and preserved only what the environment demanded. Across the deep ocean, evolution has repeated a particular calculation so many times that it begins to resemble a law. The species involved are often unrelated. Their ancestors followed different paths. Their bodies are built from different biological blueprints, yet when food becomes rare enough, they arrive at remarkably similar conclusions. Eat as much as possible when the opportunity appears, then survive whatever span of emptiness follows. The gulper eel embodies this principle with startling clarity. Its head and jaws dominate its anatomy, occupying a substantial portion of its total length. The mouth unfolds into a vast, hinged structure capable of opening far beyond what would seem proportional for the animal's size. Prey several times wider than the eel itself can be engulfed. Behind this enormous feeding apparatus, the rest of the body narrows dramatically. Muscle, tissue, and tail appear secondary, almost as though the animal was designed beginning with the mouth and completed only afterward. A similar logic appears in the black swallower. This fish rarely exceeds 25 centimeters in length, which is about 9.8 inches, yet it routinely attempts meals of astonishing scale. Its stomach stretches into a translucent chamber that can accommodate prey many times larger than its own body mass. After feeding, the swallowed animal may remain visible through the skin, its outline preserved within the expanded stomach wall. The strategy is effective often enough to persist, though it occasionally carries severe consequences. In some documented cases, the captured prey begins decomposing before digestion can be completed. Gases accumulate within the stomach, increasing buoyancy until the fish rises toward waters where survival becomes impossible. The effort to secure enough food can, on rare occasions, become fatal. Elsewhere in the darkness, the viperfish pursues the same challenge through different architecture. Its teeth are so elongated that they extend beyond the boundaries of the closed mouth. Curving upward toward the head, they resemble polished needles more than conventional fangs. Rather than relying upon repeated bites, the fish often impales prey during rapid attacks. The teeth function almost like fixed spears, securing a target before escape becomes possible. Every fraction of a second matters when opportunities are infrequent. The loose-jaw dragonfish takes a more unconventional approach. Much of the lower jaw consists of little more than a framework of bone and transparent tissue. The familiar floor of the mouth is absent. This unusual design reduces resistance as the jaw opens through the water. During an attack, less water is pushed aside. Fewer disturbances travel ahead of the predator. Prey receives less warning that danger is approaching. A seemingly fragile structure becomes an elegant solution to a problem imposed by scarcity. Not every adaptation centers on capturing food. Some focus instead on enduring its absence. The giant isopod, a distant relative of the small pill bugs found in garden soil, has transformed patience into a biological strategy. Reaching sizes vastly larger than its terrestrial relatives, it moves through the deep sea with a metabolism operating at an exceptionally restrained pace. One individual housed in a Japanese aquarium refused food for 5 years and 4 months. Throughout that period, it continued living without signs of immediate starvation. The body simply consumed energy so slowly that survival remained possible for years between meals. Seen together, these creatures reveal a recurring pattern. One lineage enlarges its jaws, another stretches its stomach, a third develops spear-like teeth, a fourth slows the passage of life itself. The details differ, yet the underlying pressure remains unchanged. Food arrives unpredictably, calories cannot be wasted, Missed opportunities may not return for a very long time. The result is a gallery of bodies reshaped around a single environmental demand. Muscles, teeth, stomachs, jaws, and metabolism have all been revised according to the same instruction. Secure nourishment whenever it appears, then endure the waiting. In the deep ocean, survival often depends less on finding abundance than on making the rarest moments of abundance last for as long as possible. There was no single moment when the deep ocean learned to glow. No ancestral species switched on a light and passed the secret to everything that followed. Instead, across vast spans of evolutionary time, dozens of unrelated lineages arrived at the same solution independently, as though separated branches of life were all solving the same problem in isolation and repeatedly arriving at the same answer. Bioluminescence, the production of visible light by living organisms, has evolved at least 40 separate times within marine environments. These were not variations inherited from a common luminous ancestor. They were independent inventions, different organisms, different histories, different anatomies, converging upon remarkably similar chemistry. Few traits in natural history demonstrate this degree of evolutionary repetition. The reason lies in the environment itself. Darkness dominates nearly every aspect of life below. When visibility vanishes, information becomes scarce. Locating food becomes difficult. Identifying members of the same species becomes difficult. Avoiding predators becomes difficult. Over and over, natural selection encountered these challenges and discovered that generating light from within the body could provide an advantage. The result spread widely. More than 76% of marine animals inhabiting the mesopelagic zone are estimated to possess some form of bioluminescence. In these waters, producing light is not unusual. It is common. Entire communities shimmer with signals, flashes, pulses, and glows generated by living chemistry. Beneath this diversity lies a surprisingly consistent mechanism. In many species, a molecule called luciferin reacts with an enzyme known as luciferase in the presence of oxygen. The reaction releases visible light. Unlike a flame, however, almost no energy is wasted as heat. The illumination is efficient, producing brightness without the costly thermal losses associated with conventional combustion. In an environment where calories are precious, such efficiency matters enormously. Once the chemistry existed, evolution began adapting it to different purposes. Some organisms use light to attract prey. Others use it to locate potential mates, drifting through immense volumes of water. Certain species communicate with flashes unique to their own lineage, allowing recognition where visual landmarks do not exist. Some release luminous material into the surrounding water when threatened, creating distractions that draw attention away from the escaping animal. Others use light as camouflage, blending with faint illumination filtering downward from above. One lineage carried the idea in an especially unusual direction. The loose-jaw dragonfish produces red bioluminescent light from organs positioned near its eyes. This ability is remarkable because most deep-sea animals cannot perceive red wavelengths effectively. Blue and green light travel farther through seawater and dominate the visual world of the deep. Red fades rapidly and becomes largely absent. The dragonfish turned that limitation into an advantage. Equipped with visual systems capable of detecting the red light it generates, it illuminates prey that remain unaware they are being observed. The effect resembles carrying a lantern whose beam is visible only to its owner. Nearby animals continue moving through darkness, unaware that they have entered another creature's field of view. Elsewhere, a different solution emerged. The gossamer worm, known as Tomopterus, produces yellow bioluminescence, a rarity among deep-sea organisms. When disturbed, it releases glowing material into the water. The luminous cloud drifts away from the worm itself, creating a moving distraction that captures attention while the animal slips elsewhere. The chemistry serves neither hunting nor communication. It functions as misdirection. What makes these examples remarkable is not merely their diversity, but their independence. Separate evolutionary histories repeatedly discovered that light could solve problems created by darkness. The same environmental pressure generated similar innovations again and again, reshaping unrelated organisms through parallel pathways. Gradually, the deep ocean became filled with living sources of illumination. Countless species carried their own signals through the water, each glow serving a purpose crafted by natural selection. Yet every new point of light introduced a consequence as well. In a world where visibility is otherwise limited, producing illumination can reveal far more than intended. A glow may attract prey, but it can also announce the location of the creature creating it. The gift of light carried a cost from the very beginning. A single flash can change everything. In the deep ocean, where many creatures manufacture their own illumination, light becomes more than a tool. It becomes information. The instant a glow appears, it announces a location, reveals movement, and creates an opportunity for another organism to interpret what it has seen. Every advantage gained through bioluminescence invites a response. Over millions of years, this exchange has unfolded again and again, producing one of the most intricate evolutionary contests on Earth. Among the participants is the hatchetfish, a small animal whose body resembles a polished blade viewed from the side. Along the underside of its body runs a series of specialized light-producing organs known as photophores. These structures emit a faint blue glow directed downward through the water. The purpose is surprisingly subtle. Even at considerable depth, traces of light filter from the distant surface above. Predators searching upward often detect prey as dark silhouettes crossing that faint illumination. The hatchetfish counters this threat by producing light that closely matches the brightness and color of the water overhead. Viewed from below, its outline begins to disappear. The body blends into the surrounding glow so effectively that it can seem to dissolve into the water itself. For a time, this strategy proved highly effective. Yet every successful adaptation creates new opportunities for natural selection to explore. Somewhere within the same vast environment, other animals began evolving ways to detect the illusion. The barrel-eye fish and the strawberry squid arrived at similar solutions despite belonging to very different lineages. Within their visual systems, evolved specialized proteins capable of detecting polarized light. Polarization is a subtle property of illumination, one often invisible to ordinary eyes. The light emitted by photophores carries patterns that differ slightly from the naturally filtered light descending from the surface. To most observers, the disguise remains convincing. To animals equipped with polarization-sensitive vision, the deception becomes easier to recognize. What appears to be empty water begins to reveal hidden shapes. The hatchetfish is no longer perfectly concealed. As predators improved their ability to detect these differences, selective pressure shifted back toward the hatchetfish. Photophore output became more refined. Brightness regulation improved. Emission patterns changed. Small adjustments accumulated across generations. In turn, visual systems continued evolving greater sensitivity. Refinements in one lineage encouraged refinements in another. The cycle repeated continuously. No participant designed the contest. No observer directed it. The process emerged naturally from the interaction between light and biology. Across millions of years, countless tiny modifications accumulated, each preserving a slight advantage until another species discovered a way around it. The result resembles an arms race conducted through optics rather than weapons. Not every lineage chose camouflage as its solution. Some approached the problem from a completely different direction. Instead of hiding the light they produced, they focused on eliminating reflected light altogether. If illumination from another organism struck their bodies, almost none of it would return to the surrounding water. This strategy appears in several species of ultrablack fish. Their skin absorbs extraordinary amounts of incoming light, preventing reflections that might otherwise reveal their presence. In an environment increasingly crowded with flashes, glows, and biological lanterns, the ability to swallow light becomes a powerful form of concealment. A passing burst of bioluminescence may illuminate nearby water. It may strike rocks, suspended particles, or the bodies of other animals. Yet, when it reaches ultrablack skin, almost nothing returns. The fish remains hidden not because it has matched the environment, but because it has removed itself from the visual equation as completely as possible. Taken together, these adaptations reveal how profoundly bioluminescence transformed deep ocean life. Once creatures began creating light, every organism around them had to respond. Some learned to imitate ambient illumination. Others learned to detect subtle flaws in that imitation. Still others abandoned the contest entirely and focused on becoming nearly invisible to any light that touched them. The spread of living illumination did not simplify life in the deep ocean. It made the environment more complex, turning every flash into both opportunity and risk, and every glow into something that could be used, interpreted, concealed, or betrayed. Imagine a flash of living light crossing the water. For an instant, the surrounding darkness is interrupted. A drifting crustacean becomes visible. The outline of a predator emerges. Tiny particles suspended in the water shimmer and disappear again. Then the light reaches the skin of another fish, and something unusual happens. Almost none of it returns. In the deep ocean, where illumination is scarce and often biological in origin, reflection can be dangerous. Every returning particle of light carries information. It reveals shape, position, direction, and movement. Across millions of years, this simple reality exerted extraordinary pressure on certain species, gradually producing one of the most remarkable optical materials known in nature. In 2020, researchers at the Smithsonian National Museum of Natural History documented 16 species of deep-sea fish possessing skin of exceptional darkness. Measurements revealed that some reflected as little as 0.05% of incoming light. To appreciate the significance of that figure, comparison becomes useful. Black velvet reflects roughly 0.5%. Specialized coatings developed for stealth aircraft reflect around 0.1%. These fish, relying entirely upon biological processes, approached and in some cases exceeded levels of darkness associated with highly advanced human engineering. At first glance, the explanation might seem straightforward. Many animals appear black because they possess dark pigments. Yet the mechanism operating within these fish is far more refined. Their skin contains dense concentrations of structures known as melanosomes, microscopic compartments filled with melanin pigment. Melanin is common throughout the living world. It colors feathers, fur, skin, scales, and countless other tissues. What distinguishes these deep-sea species is not the pigment itself, but the arrangement. The melanosomes are packed together with extraordinary precision. When light enters the skin, some photons are absorbed immediately. Those that escape initial absorption encounter neighboring melanosomes, positioned in such a way that the photons are redirected sideways rather than outward. Each deflection increases the likelihood of another encounter with pigment. Again and again, the light is passed from structure to structure until absorption becomes inevitable. The process functions as a microscopic maze designed specifically for photons. Light enters, light wanders, light disappears. One researcher described the effect in simple terms. The light does not bounce back. It does not pass through. It enters the layer and vanishes. Beneath that simplicity lies an elegant piece of biological engineering shaped entirely through natural selection. Many other dark-colored animals rely on additional structural materials to enhance absorption. Feathers, scales, and external matrices help trap incoming light before it can escape. Deep-sea ultrablack fish accomplish something even more efficient. They achieve extraordinary darkness using densely organized melanosomes alone, without requiring extensive supporting structures. The result is a thinner and less resource-intensive system. That efficiency matters. Every biological structure carries a cost. Tissue requires energy to build and maintain. In environments where calories arrive unpredictably, unnecessary expenditures become liabilities. Evolution favors solutions that accomplish more with less. The advantage of ultra-black skin becomes clear whenever bioluminescence illuminates the surrounding water. A passing glow may sweep across the body of a fish. If even a small fraction of that light reflects outward, nearby predators may detect it. Prey may recognize approaching danger. The encounter changes. When absorption reaches 99.95%, instead of 98%, the difference is not aesthetic. It is functional. The animal becomes dramatically harder to detect. A body that once reflected a recognizable outline begins dissolving into the background. The effectiveness of this strategy has attracted attention beyond marine biology. Engineers designing advanced optical systems, particularly those used in telescopes, have begun studying ultra-black fish skin as a source of inspiration. Instruments intended to detect faint, distant objects benefit from surfaces that absorb stray light as completely as possible. In this way, a solution developed over evolutionary timescales in the deepest ocean now informs technological challenges far removed from the water. Long before laboratories attempted to create perfect darkness, certain fish had already arrived there through countless generations of refinement, transforming their skin into one of nature's most effective traps for light. Not every creature responded to darkness by seeing more. Some responded by seeing less. Across the deep ocean, vision was pulled in opposite directions by the same environmental force. In certain lineages, eyes gradually diminished until little remained. In others, they expanded into structures of astonishing scale and complexity. The divergence reveals something fundamental about evolution. There is rarely a single solution to a problem. There are only solutions that fit the circumstances of a particular organism at a particular moment. For some animals, maintaining vision simply ceased to be worthwhile. Eyes are expensive organs. They require specialized neural tissue to process incoming information. They demand a constant supply of nutrients and oxygen. They occupy space that could otherwise be used for other biological functions. When an environment provides little useful visual information, these costs begin to outweigh the benefits. Over immense spans of time, Numerous deep-sea species reduced their visual systems accordingly. Eyes shrank, lenses simplified, neural pathways associated with vision diminished. In certain lineages, the structures became little more than vestiges, faint reminders of ancestors that once inhabited brighter waters. In a realm where photons rarely arrive, preserving the machinery designed to detect them can become an unnecessary burden. Yet elsewhere, evolution moved with equal determination in the opposite direction. The colossal squid possesses the largest eyes known in any living animal. Each eye reaches approximately 27 centimeters in diameter, which is about 10.6 inches. Such dimensions would appear excessive in almost any other environment. These eyes were not shaped for reading fine details or distinguishing intricate patterns. Their purpose is simpler and, in some ways, more profound. They are designed to detect the presence of light itself. A distant flash produced by bioluminescent organisms may signal movement. It may reveal prey. It may indicate the approach of a predator large enough to disturb entire communities of glowing animals as it passes. The larger the eye, the greater its ability to gather scarce photons. In darkness, sensitivity often matters more than clarity. The giant isopod arrived at a different variation of the same principle. Its compound eyes contain nearly 4,000 individual facets. Rather than maximizing resolution, this arrangement broadens coverage. Light or movement approaching from many directions can be detected simultaneously. The visual world remains sparse, but awareness extends across a wider field. Then there is the barrel-eye fish, an animal whose true appearance remained hidden from science for decades. Researchers collected specimens for many years and examined them carefully, yet every individual brought to the surface carried the same problem. The dramatic pressure change damaged delicate structures before scientists could observe them intact. A transparent dome covering the head collapsed during retrieval, leaving behind a distorted version of the living animal. As a result, generations of researchers unknowingly studied an incomplete picture. Features believed to be eyes were actually olfactory organs. The genuine visual system remained concealed within the damaged dome. The misunderstanding persisted until 2009, when researchers from the Monterey Bay Aquarium Research Institute recorded living barrel-eye fish in their natural environment using a remotely operated vehicle. For the first time, the animal appeared as it truly existed beneath the water. Inside the transparent dome sat two vivid green tubular eyes. Rather than remaining fixed, these eyes could rotate within the skull. They often pointed upward, scanning for silhouettes drifting against the faint illumination filtering from above. When prey was detected, the eyes rotated forward, allowing more precise tracking during an approach. The green pigmentation surrounding the visual system serves an additional purpose. It filters incoming light, reducing interference from residual sunlight while allowing bioluminescent wavelengths to pass more effectively. The result functions as a biological optical filter, refined through evolutionary time to emphasize the signals most relevant to survival. Seen in this context, the barrel-eye fish appears less like an oddity and more like a specialized instrument. Its transparent dome, rotating eyes, and selective filters form a system engineered by natural selection for a world where information arrives only in fleeting traces. For decades, science misunderstood the design because it encountered the animal only after the environment that shaped it had already been stripped away. Far beneath the broad expanses of the open ocean, the deepest parts of Earth are not arranged as one connected realm. They are scattered, separated, and isolated, hidden within narrow geological wounds that descend into the planet's crust. From above, they appear as lines on a map. From within, they are worlds unto themselves. These regions form the hadal zone, named after Hades, the ancient underworld. Yet the hadal zone is not a single place. It consists of 37 distinct trenches distributed across the global ocean. Most lie around the Pacific Ring of Fire, the immense tectonic boundary where sections of Earth's crust converge and one plate gradually sinks beneath another. Over millions of years, this process carves steep, elongated depressions into the seafloor, creating some of the deepest environments on the planet. The trenches vary greatly in depth. The Middle American Trench reaches approximately 6,669 meters, which is about 4,144 miles. The Mariana Trench descends to roughly 10,989 meters, which is about 6,827 miles. Between these extremes lies a diverse collection of other trenches, each possessing its own geography, chemistry, and biological community. What makes these environments particularly remarkable is their separation from one another. Vast abyssal plains stretch between neighboring trenches. These plains may appear featureless, but for many hadal organisms they function as nearly impassable barriers. Species adapted to one trench often cannot travel across thousands of kilometers, which is about 1,864 miles of deep seafloor, to reach another. As a result, every trench behaves much like an island. The comparison becomes even more striking when viewed through the lens of evolution. On islands, populations become isolated. Over time, they follow independent paths. The same process unfolds in the hadal zone, except the isolation occurs beneath immense pressure and perpetual darkness rather than above sea level. Each trench operates as a sealed biological laboratory. Life enters, adapts, diversifies, and changes without meaningful contact with neighboring systems. For millions of years, these experiments have proceeded independently. Organisms in one trench respond to local conditions. Organisms in another trench respond to slightly different circumstances. Evolution explores possibilities separately in each location. Even within a single trench, boundaries emerge. Around 6,000 meters, which is about 3,728 miles, researchers observe a notable transition in community composition. Species common at shallower depths begin disappearing. New organisms emerge in their place, many found nowhere else on Earth. Descending farther produces additional changes. The animals inhabiting 7,000 meters, which is about 4,349 miles, often differ from those living at 9,000 meters, which is about 5,592 miles. Depth itself becomes a form of geographic separation. Despite their significance, these environments remain among the least visited places on the planet. Challenger Deep, the deepest known location within the Mariana Trench, has received remarkably few human visitors. In 1960, Jacques Piccard and Don Walsh descended there aboard the bathyscaphe Trieste. After that historic journey, more than half a century passed before another crude descent occurred. In 2012, James Cameron reached the site alone. 52 years separated those visits. The interval illustrates the immense difficulty of exploring such depths. Reaching the hadal zone requires extraordinary engineering, extensive resources, and a willingness to operate in conditions that challenge both machinery and human endurance. Many regions have never been directly observed by people at all. Meanwhile, the evolutionary experiments continued uninterrupted. Generation followed generation within isolated trenches scattered across the Pacific and beyond. Separate populations encountered similar pressures, similar limitations, and similar opportunities. For millions of years, each trench refined its own solutions beneath the weight of the ocean. When scientists eventually began comparing the outcomes emerging from these isolated worlds, the patterns they discovered hinted at something deeply unexpected. Independent evolutionary histories, separated by vast distances and immense barriers, had begun arriving at remarkably similar destinations. Separated by immense distances, the trenches of the hadal zone might be expected to produce entirely different forms of life. Each exists as its own isolated world. Each possesses its own geological history. Each has spent millions of years developing largely apart from the others. Yet when biologists compare the creatures emerging from these distant environments, a striking pattern begins to appear. Again and again, life arrives at familiar answers. The repetition is not exact. Species remain unique. Their evolutionary histories differ. The molecular pathways guiding adaptation are often unrelated. Yet the resulting designs frequently resemble one another in ways that are difficult to dismiss as coincidence. Across multiple trenches, organisms have evolved bodies rich in water and low in rigid structural material. Gelatinous tissues appear repeatedly because they tolerate immense pressure more effectively than heavily reinforced frameworks. Swim bladders, useful for buoyancy control in shallower waters, vanish from many hadal species because gas-filled structures become increasingly impractical at extreme depths. Metabolism slow, growth rates decline, lifespans lengthen, feeding structures expand, pigmentation often diminishes or disappears altogether. Similar patterns emerge across lineages that share no recent ancestry. The deeper researchers look, the more examples accumulate. Dragonfish and viperfish belong to separate evolutionary branches, yet both developed elongated fang architectures capable of securing prey during brief encounters. The details differ, but the functional outcome remains remarkably similar. In another example, multiple unrelated species evolved counter-illumination organs positioned in nearly identical locations along the body. The placement allows emitted light to blend with illumination arriving from above, reducing the visibility of the animal from below. Different evolutionary histories converged on nearly the same arrangement. Even pressure adaptation demonstrates this recurring tendency. Several unrelated groups evolved water-rich tissues capable of compensating for crushing depths. The molecular mechanisms are not always identical. Different biological pathways were recruited, different genetic changes accumulated, yet the destination remained similar. Separate routes led toward the same solution. Bioluminescence provides perhaps the most dramatic illustration. As explored earlier, the ability to generate light evolved independently, at least 40 times. Such repetition suggests that certain environmental challenges consistently favor particular responses. When unrelated organisms repeatedly discover the same strategy, the environment itself begins to look less like a backdrop and more like an active sculptor. Evolutionary biologists refer to this phenomenon as convergent evolution. The concept carries a profound implication. Some traits arise because of unique historical circumstances. Others emerge because the laws governing an environment leave relatively few viable alternatives. The hadal zone appears to belong strongly to the latter category. Pressure, darkness, scarcity, and cold act together to restrict biological possibilities. Many potential body plans simply do not function under such conditions. Certain adaptations become advantageous repeatedly because physics rewards them repeatedly. Over time, natural selection filters possibilities until unrelated species begin resembling one another in function, even when they remain distant in ancestry. Viewed from this perspective, the creatures inhabiting the deepest ocean are not random outcomes. They are responses generated by a specific set of environmental constraints. Given sufficient time, evolution explores countless variations, yet many pathways eventually converge upon familiar themes. The idea carries an unusual sense of inevitability. If sunlight is removed, if food becomes scarce, if pressure remains immense and unrelenting, and if these conditions persist for hundreds of millions of years, certain biological solutions become increasingly probable. Enlarged feeding structures appear, metabolisms slow, specialized pressure adaptations emerge. Light is produced, manipulated, hidden, and detected. Different organisms discover these strategies independently because the environment continually favors them. In this way, the deepest ocean reveals something broader about life itself. Evolution is creative, but it does not operate without limits. The physical world imposes boundaries, and within those boundaries, certain designs become remarkably likely. Across isolated trenches scattered around the planet, nature has demonstrated this principle repeatedly, producing similar answers from separate beginnings. The pattern suggests that some forms are not merely possible. Under the right conditions, they may be expected. The discovery began not with biologists searching for life, but with geologists searching for heat. In February of 1977, aboard the research vessel Canour, a scientific team worked above a remote portion of the Pacific seafloor known as the Galapagos Rift. The region lay along a tectonic boundary where Earth's crust slowly pulled apart. Beneath the seabed, molten material rose from deeper layers of the planet, warming the surrounding rock. The researchers expected signs of geological activity, perhaps warm water escaping through cracks in the ocean floor. Nothing in their plans suggested they were approaching one of the most important biological discoveries of the 20th century. As instruments descended into the darkness, observations began returning from depths where sunlight had never penetrated. The water was cold, hovering near freezing. Pressure remained immense. By every conventional expectation, this should have been a place of scarcity. Instead, the seafloor appeared crowded with life. Dense clusters of organisms surrounded openings where mineral-rich water surged upward from beneath the crust. Giant tube worms stood in pale forests. Large clams gathered across the sediment. White crabs moved among rocky outcrops. Strange shrimp occupied the margins of the vents. Bacterial mats spread across surfaces in shades of rust, orange, and white. The scene was not merely inhabited, it was thriving. At the time, this presented a profound problem for biology. For generations, scientists had worked from a foundational assumption that seemed almost self-evident. Every ecosystem on Earth ultimately depended upon sunlight. Trace any food chain backward through enough links, and eventually it would arrive at photosynthetic organisms. Plants, algae, and microscopic producers captured solar energy and converted it into organic matter. Herbivores consumed those organisms. Predators consumed herbivores. The details varied, but the source appeared universal. The assumption had become so deeply embedded that it often went unstated. Yet here, thousands of meters beneath the surface, which is about thousands of miles below the realm reached by sunlight, abundant life flourished where photosynthesis was impossible. No sunlight reached these depths. No plant could grow there. No algae could harvest energy from the sky above. The ecosystem existed anyway. The explanation emerged through careful study. The vents released water enriched with dissolved chemicals from deep within Earth's crust. Among these chemicals was hydrogen sulfide, a compound toxic to many surface organisms. For certain bacteria, however, hydrogen sulfide represented an energy source. These microorganisms performed chemosynthesis. Rather than using sunlight to construct organic molecules, they used chemical energy extracted from vent fluids. The process differed from photosynthesis in mechanism, but it achieved a similar outcome. Energy became living matter. Those bacteria formed the foundation of an entire food web. Tube worms developed intimate partnerships with chemosynthetic bacteria living inside their bodies. Clams hosted similar microbial communities. Countless other organisms depended directly or indirectly upon this chemical productivity. An ecosystem of remarkable complexity operated entirely on energy rising from within the Earth itself. Members of the expedition later described their astonishment. One researcher recalled that the team was completely surprised. The reaction was understandable. According to prevailing expectations, such a community should not have existed, yet there it was, flourishing in plain view on the ocean floor. The discovery transformed scientific understanding almost immediately. As exploration expanded, hydrothermal vent systems began appearing elsewhere. Researchers identified active vent fields throughout the world's oceans, from polar waters in the north to polar waters in the south. More than 500 active hydrothermal vent regions are now known. Each functions as a self-contained ecosystem fueled primarily by chemistry rather than sunlight. These communities demonstrated that life could persist under conditions previously considered unsuitable for complex ecological networks. Energy need not arrive from above. Under the right circumstances, it could emerge from below. The implications extended far beyond marine biology. A principle once regarded as nearly universal had required revision. The requirements for a functioning ecosystem proved broader than scientists had imagined. In the darkness surrounding hydrothermal vents, life revealed that it possessed more pathways than the old rulebook allowed, and the definition of what an ecosystem needed to survive had to be written anew. Among the many inhabitants of hydrothermal vent ecosystems, few challenge familiar ideas about animal life as completely as the giant tube worm. At first glance, it appears straightforward, a long, pale tube anchored to the seafloor and crowned with a vivid red plume extending into the surrounding water. Yet beneath that simple exterior exists a biological arrangement so unusual that it reshapes the very definition of what it means for an animal to feed itself. The species Riftia pachyptyla can reach lengths of approximately 2.4 meters, which is about 7.9 feet. It is also among the fastest growing marine invertebrates known to science, capable of adding as much as 85 centimeters of length within a year, which is about 2.8 feet. Such rapid growth would be impressive under any circumstances. Within the harsh environment of hydrothermal vents, it becomes even more remarkable. What makes the tube worm truly extraordinary, however, is not its size or growth rate. It has no mouth. It possesses no stomach, no intestines, and no digestive tract. The anatomical structures that animals typically rely upon to consume and process food are entirely absent. Throughout its evolutionary history, this species never developed the machinery required for eating in the conventional sense. The omission appears impossible at first, yet the worm thrives precisely because it replaced digestion with something else. Occupying much of its internal body cavity is a specialized organ known as the trophosome. Richly supplied with blood vessels, This structure serves as the living center of the worm's nutritional system. Within it reside billions of chemosynthetic bacteria. These microorganisms perform the biochemical work the worm itself cannot accomplish. Hydrothermal vents release hydrogen sulfide into the surrounding water, a compound toxic to many forms of life. The bacteria use that chemical as an energy source. Through chemosynthesis, they convert inorganic compounds into organic material, producing nutrients that the worm absorbs directly through its tissues. The partnership is astonishingly intimate. The bacteria receive shelter, stability, and a steady supply of raw materials. The worm receives nourishment. Neither partner performs its role independently. Together, they create a functioning biological system greater than either component alone. The bright red plume extending from the top of the worm's tube serves as the interface connecting this partnership to the outside world. Richly supplied with blood, the plume extracts oxygen and hydrogen sulfide from the surrounding water. Ordinarily, these substances would react in ways that complicate transport through the bloodstream. The tube worm solves this challenge through a highly specialized form of hemoglobin. Unlike any other known hemoglobin, it can carry both oxygen and hydrogen sulfide simultaneously without allowing them to react with one another. This adaptation permits the worm to gather the precise ingredients required by its bacterial partners and deliver them safely to the trophosome deep within its body. As researchers examined hydrothermal vent ecosystems across the world's oceans, another pattern began to emerge. The tube worm's strategy was not unique. Giant vent clams evolved symbiotic relationships with chemosynthetic bacteria living within their tissues. Vent mussels developed comparable arrangements. Several unrelated worm species arrived at similar solutions despite following different evolutionary histories. Again and again, animals confronting the challenges of chemical ecosystems discovered the same fundamental approach. Rather than performing all biochemical processes themselves, they delegated crucial metabolic functions to microbial partners. This repetition echoes a theme that has appeared throughout the deep ocean. Independent lineages repeatedly encounter the same environmental pressures and arrive at remarkably similar outcomes. Convergent evolution extends beyond body shape, feeding structures, or sensory systems. In hydrothermal vent communities, it reaches all the way down to metabolic architecture itself. The giant tube worm represents perhaps the clearest expression of that principle. It is not merely an animal hosting bacteria, nor is it simply a colony of microbes living inside an animal. The boundaries separating the two become increasingly difficult to define. Nutrients, chemicals, and biological functions pass continuously between partners until the distinction begins to blur. What emerges is a single integrated system assembled from two different forms of life, each performing tasks the other cannot, functioning together with such completeness that neither can truly be understood in isolation. At the greatest depths, where a vertebrate has ever been observed, elegance takes a form that is easy to misunderstand. In 2023, researchers operating a deep ocean camera system within the Ogasawara Trench recorded a small snailfish at a depth of 8,336 meters, which is about 5,180 miles. The fish was not hiding among rocks or drifting cautiously through the water. It approached the bait directly, moving with an ease that seemed almost casual despite existing under conditions that would destroy most forms of vertebrate life. The deepest fish ever physically collected belongs to a species known as Pseudoliparis swirei, commonly called the Mariana snailfish. Retrieved from approximately 8,022 meters, which is about 4,985 miles. It measures roughly the length of a human forearm. For an animal occupying the most extreme depths available to any known fish, its appearance can be surprisingly understated. There are no oversized fangs, no glowing lure extends from its head, no protective armor plates cover its body. Nothing about its appearance resembles the dramatic imagery often associated with deep ocean predators. Instead, it is small and pale. Its body is partially translucent, scales are absent, pigmentation is minimal. In some specimens, internal organs can be seen through the body wall. The liver appears faintly visible beneath the skin. The bones are thin and lightly mineralized. Portions of the skull remain incompletely fused. Viewed through the expectations formed by life at the surface, the animal can seem unfinished. Yet every apparent imperfection is the result of extraordinary refinement. At depths exceeding 8,000 meters, survival depends less on adding structures than on removing them. Evolution has spent millions of years eliminating anything that creates unnecessary stress within an environment dominated by immense pressure. Scales provide a useful example. In shallower habitats, they offer protection and structural support. In the hadal zone, rigid external structures can create localized pressure stresses. Over evolutionary time, the simplest solution was not to strengthen the scales, but to dispense with them altogether. Pigmentation followed a similar path. Producing pigments requires energy and biological resources. In perpetual darkness, coloration offers little practical benefit. As generations passed, reduced pigmentation became increasingly advantageous. What remained was only the minimum required by the animal's biology. The skeleton underwent its own transformation. Surface vertebrates often rely upon heavily ossified bones capable of resisting substantial mechanical forces. At hadal depths, excessive rigidity can become a liability. Softer, partially ossified structures distribute compression more effectively. Rather than resisting environmental pressure directly, the skeleton accommodates it. Even the skull reflects this principle. A completely sealed and rigid cranium may experience uneven stresses under extreme conditions. The snailfish retains a more flexible arrangement, allowing pressure to be distributed across the structure with greater uniformity. What appears incomplete is actually adaptive. Gas-filled organs present another challenge. Swim bladders function effectively in many fishes by regulating buoyancy through contained gas. Under hadal pressures, such spaces become impractical. The Mariana snailfish abandoned the strategy entirely. Instead, buoyancy is achieved through tissues rich in water and gelatinous material. Water resists compression far more effectively than gas, making it a dependable structural component in the deepest ocean. Its earliest stages of life reveal the same pattern of specialization. The species produces unusually large eggs compared with those of many shallow water relatives. There is no protected nursery above, no gradual transition into the pressures of the hadal environment. From the moment young snailfish emerge, they must function within conditions approaching the limits of vertebrate existence. Larger eggs provide greater developmental resources, allowing juveniles to begin life already prepared for that challenge. The lineage traces its ancestry to fishes that once inhabited far shallower waters approximately 20 million years ago. Across that immense span of time, natural selection performed a meticulous process of subtraction and refinement. Structures that introduced vulnerability disappeared. Traits that improved survival persisted. The result is an animal that often appears fragile to human eyes. Yet fragility is an illusion created by unfamiliar conditions. The Mariana snailfish represents one of evolution's most precise achievements, a vertebrate shaped not by excess, but by relentless optimization, retaining only what the deepest ocean allows and rewarding only what the deepest ocean requires. Size, in the deep ocean, is not a measure of dominance. It is often a measure of patience. Far below the familiar rhythms of the surface, where meals may arrive separated by weeks, months, or longer spans of time, evolution follows a different arithmetic. Growth is no longer balanced against abundance. Instead, every trait is weighed against scarcity, and in many lineages, that calculation has produced creatures whose proportions seem almost impossible when compared with their relatives elsewhere on Earth. Consider the giant isopod, Bathynomus giganteus. Its distant relatives are familiar to many people, small pill bugs that wander through garden soil and beneath fallen leaves. Those terrestrial forms typically reach lengths of around 2 centimeters. The giant isopod, by contrast, can grow to approximately 50 centimeters, which is about 1.6 feet, and may weigh as much as 1.7 kilograms. The relationship between the two is unmistakable. The segmented armor, the curved body, the overlapping plates all reveal a shared ancestry. Yet one lineage remained small, while the other expanded into something nearly 25 times larger. A similar pattern appears among amphipods. Near the surface, these crustaceans are often measured in mere millimeters, tiny drifting components of marine food webs. In the Mariana Trench, however, researchers have collected supergiant amphipods exceeding 34 centimeters in length, which is about 1.1 feet. The body plan remains recognizable, but the scale has shifted dramatically, as though the environment itself stretched familiar designs into unfamiliar dimensions. The trend extends beyond crustaceans. Among cephalopods, the giant squid reaches lengths of approximately 13 meters, which is about 42.7 feet. The colossal squid, even heavier and more robust, has been measured at more than 700 kilograms. Its eyes can approach 27 centimeters in diameter, which is about 0.9 feet. These immense organs are not luxuries. They are tools shaped by a world where detecting the faintest signal may determine whether a meal is found or missed. What makes these examples remarkable is not their individual scale, but their repetition. Deep-sea gigantism appears across crustaceans, cephalopods, worms, and numerous other groups. The pattern emerged independently many times, suggesting that the environment repeatedly favors the same outcome. The reasons are woven together through the physics of cold water and the economics of limited energy. Lower temperatures, slow metabolism. Organisms process resources more gradually, grow more slowly, and often live longer lives. Extended lifespans create opportunities for continued growth across many years before reproduction becomes the primary biological priority. At the same time, larger bodies possess characteristics that can be advantageous in a resource-constrained environment. As size increases, volume grows more rapidly than surface area. This relationship improves the efficiency with which an organism retains the energy generated by its metabolism. Each unit of body mass requires proportionally fewer calories for maintenance. When food is unpredictable, those savings become significant. A larger animal can also endure longer intervals between meals. Energy reserves remain available for extended periods, reducing dependence upon frequent feeding opportunities. In habitats where edible material may drift downward only occasionally, endurance can be more valuable than speed. Yet gigantism is not universal. The same ocean that enlarged the giant isopod reduced the male anglerfish to a fraction of the female's size. The same darkness that produced colossal squid also shaped delicate snailfish adapted to entirely different constraints. No single body plan dominates. No preferred scale governs every lineage. The deep ocean rewards efficiency, and efficiency takes many forms. For some creatures, survival meant becoming smaller, lighter, and simpler. For others, it meant continuing to grow, storing energy within larger bodies capable of enduring long stretches of scarcity. Both strategies emerged from the same environment, guided by the same pressures, arriving at opposite results. In that sense, the giants of the deep are not exceptions to the rules of their world. They are expressions of those rules, shaped by cold water, limited food, and immense spans of time into forms that appear extraordinary only because the calculations that created them unfolded far beyond the reach of sunlight. Some evolutionary stories are written through addition. New structures appear, new capabilities emerge, and bodies grow more elaborate across time. Yet in vast regions of the deep ocean, a different pattern unfolds. Generation after generation, natural selection removes rather than adds, trimming away anything that consumes energy without providing sufficient return. The result is a collection of organisms that seem less constructed than distilled, reduced to forms that approach the smallest possible biological investment compatible with survival. Transparency is one of the most successful expressions of that process. Across the mesopelagic and upper bathypelagic realms, countless lineages have independently arrived at the same solution. Glass squid drift through the water with bodies that seem scarcely present at all. Larval eels resemble floating strands of crystal. Transparent shrimp carry their organs within nearly invisible shells. Comb jellies and gelatinous drifters move through the water as if assembled from faint reflections and liquid geometry rather than flesh. In a world where bioluminescent flashes provide much of the available illumination, visibility becomes a dangerous currency. Any surface capable of reflecting light may reveal its owner. Any pigmentation may create a detectable outline. Transparency offers an elegant alternative. A body that neither strongly reflects nor strongly absorbs incoming light becomes extraordinarily difficult to perceive. It occupies space physically while surrendering much of its visual presence. Achieving this state requires far more than simply removing pigment. Light interacts with tissues in complicated ways. Muscle fibers, connective structures, and internal organs can all scatter illumination, producing subtle distortions that reveal an organism's location. Deep-sea animals that rely on transparency often possess tissues arranged with remarkable precision, minimizing the optical disruptions that would otherwise betray them. Internal anatomy presents an especially difficult challenge. An opaque organ enclosed within a transparent body can create a distinct silhouette. Evolution has therefore favored multiple solutions. In some species, organs are compressed into narrow arrangements that reduce their visibility. In others, particular tissues are darkened selectively, restricting reflected light to small regions while preserving transparency elsewhere. Every adjustment serves the same objective: reducing the body's visual footprint within an environment where detection carries consequences. Among gelatinous organisms, this pursuit of efficiency extends beyond optics. Many deep-sea jellyfish and comb jellies consist of more than 95% water by mass. At first glance, such construction appears delicate. From an energetic perspective, however, it is profoundly effective. Water is already abundant in the surrounding environment. Incorporating it into body structure requires far fewer biological resources than building dense layers of muscle or supportive tissue. The resulting forms possess very little solid material. Their architecture approaches a minimum threshold, enough structure to move, feed, reproduce, and persist, while avoiding unnecessary investment. They are not incomplete organisms. They are examples of extraordinary metabolic economy. A similar principle appears in the Dumbo octopus. Unlike many shallow water octopuses, it relies less upon energetic bursts of jet propulsion. Instead, it moves through the water using gentle fin strokes that resemble the motion of wings. This mode of travel demands far less energy over time. Another structure has also faded from importance. The ink sac, a valuable defensive feature in illuminated waters, offers little advantage in darkness, where visual concealment through ink clouds becomes largely ineffective. In the Dumbo octopus, that organ has been reduced to vestigial tissue. Again and again, the same lesson emerges. The deep ocean does not reward complexity for its own sake. It evaluates every structure according to cost. Every gram of tissue must justify its existence. Every organ must earn the energy required to maintain it. The animals that thrive here are therefore not primitive remnants lingering from earlier evolutionary stages. They represent highly specialized outcomes shaped through immense spans of time. Their bodies have been refined by subtraction, stripped of excess, until only the most valuable functions remain. What appears simple from a surface perspective is often the product of astonishing precision. Transparency, gelatinous construction, reduced organs, and minimalist movement all reflect the same underlying principle. In a realm where resources are limited and efficiency governs survival, life often advances not by becoming more elaborate, but by discovering how little it can afford to be while remaining alive. Far above the abyssal floor, a whale's life may span decades as it crosses entire ocean basins. Yet one of its most profound ecological contributions begins only after that life has ended. When a great whale dies in the open ocean, the body begins a long descent through layers of water that have shaped so many of the creatures encountered in this journey. The fall may take several hours, or in some cases, several weeks, depending on currents, buoyancy, and the condition of the carcass. As it sinks, it carries with it an immense cargo of stored biological energy. A large blue whale can contain as much as 100 metric tons of organic matter, an extraordinary concentration of nutrients entering a realm where food is often dispersed as tiny fragments drifting downward from distant surface waters. Eventually, the body reaches the seafloor. For the surrounding inhabitants, the arrival resembles the sudden appearance of an oasis within a vast desert. What may have been an expanse of sparsely distributed resources becomes the site of intense biological activity. The transformation begins almost immediately. The first phase is known as the mobile scavenger stage. Within hours, animals capable of detecting the new resource begin to gather. Sleeper sharks arrive. Hagfish thread their way into soft tissues. Rat-tail fish and numerous crustaceans join the growing assemblage. Feeding occurs on a scale rarely encountered in the deep ocean. Flesh that once powered migrations across thousands of kilometers, which is about 1,864 miles, is rapidly converted into nourishment for an entire community. During the months that follow, as much as 90% of the whale's soft tissue may be consumed. The density of life surrounding the carcass becomes remarkable, especially when contrasted with the ordinarily dispersed nature of deep-sea ecosystems. Creatures emerge from surrounding darkness and concentrate around a single location, drawn together by a resource too valuable to ignore. As the larger scavengers exhaust the most accessible tissues, the ecosystem enters a second phase known as the enrichment opportunist stage. The whale's remains continue to influence the surrounding environment. Small fragments of organic material settle into nearby sediments. Nutrients seep outward. New groups of organisms colonize the enriched seafloor, feeding upon remnants left behind during the initial feeding frenzy. The community changes, but the flow of energy continues. Then another transformation begins, one that echoes discoveries made at hydrothermal vents. Deep within the whale's skeleton lie substantial reserves of lipids stored in the bones. Specialized bacteria begin breaking down those reserves through chemical processes that release hydrogen sulfide. Gradually, the bones become the foundation of an entirely different ecosystem. Chemosynthetic bacteria establish themselves around the skeleton. Pale bacterial mats spread across surfaces. Tube worms appear. Clams settle nearby. Mussels and specialized crustaceans join the growing assemblage. Without volcanic activity and without a hydrothermal vent, a chemically powered community emerges upon the remains of a single animal. This phase, known as the sulfophilic stage, can endure for astonishing lengths of time. Depending upon the size of the whale and local environmental conditions, the ecosystem may persist for between 50 and 100 years. A single carcass can support more than 400 species over a period longer than many human lifetimes. Some organisms have become so specialized that they exist nowhere else. More than 120 species have been documented exclusively at whale falls, their entire evolutionary history tied to the precise chemical conditions created by decomposing whale skeletons resting on the abyssal floor. Their existence depends upon events that occur only when one of the ocean's largest animals completes its final descent. Researchers estimate that approximately 690,000 whale carcasses currently rest across the seafloor of Earth's oceans. Scattered through every major basin, they form a hidden network of nutrient islands separated by immense distances, yet connected through the movements of life that discovers and inhabits them. What begins as a death becomes a century of transformation. Flesh becomes community, bone becomes habitat, chemistry becomes ecology. And across the deep ocean floor, countless generations of organisms continue living within worlds assembled from the remains of giants that vanished long ago. Across the abyssal floor, distance is often the greatest barrier life must overcome. A hydrothermal vent may flourish with dense communities of worms, clams, mussels, and microorganisms. Yet, beyond the immediate boundaries of that oasis stretches an immense expanse of seafloor where resources are sparse and opportunities for settlement are rare. For many organisms, crossing such territory presents a challenge measured not merely in space, but in generations. A larva drifting away from one productive habitat may never encounter another before its brief developmental window closes. For a long time, this presented a puzzle. Chemosynthetic ecosystems appeared isolated from one another, scattered across ocean basins like distant islands. Yet, genetic studies revealed relationships that suggested a deeper connection. Populations separated by vast distances still carried traces of shared ancestry. Somewhere within the deep ocean, pathways existed. The answer may have been resting on the seafloor all along. Along major whale migration routes, researchers estimate that the average spacing between whale falls can be approximately 12 kilometers, which is about 7.5 miles. In the most heavily traveled corridors, the the distance may shrink to only 5 kilometers, which is about 3.1 miles. Measured against the scale of the open ocean, those separations are surprisingly small. Many deep-sea larvae are capable of drifting along cold bottom currents across distances comparable to those intervals. A larva released from one whale fall may settle at another. The next generation may continue the journey, then another generation after that. What appears impossible as a single voyage becomes achievable through a sequence of smaller steps extending across centuries and millennia. The process resembles a chain of stepping stones extending through darkness. Each whale fall functions as a temporary habitat, but also as a waypoint. Organisms establish populations, reproduce, and release offspring into surrounding waters. Those offspring drift outward in search of new opportunities. If another carcass lies within reach, colonization becomes possible. Over immense spans of time, lineages can gradually expand across regions that would otherwise remain inaccessible. Evidence supporting this idea emerged through molecular studies of deep-sea animals. Mussels inhabiting hydrothermal vents show ancestral relationships with species associated with whale falls. Similar patterns appear among clams and worms. Organisms found in geographically separated vent systems often share evolutionary histories consistent with repeated movement through intermediate habitats. Whale falls appear to have served as those intermediaries. The implication is remarkable. The hydrothermal vent ecosystems that transformed scientific understanding during the discoveries of the late 20th century may never have been as isolated as they seemed. Their long-term persistence may have depended upon biological corridors created not by volcanic activity or tectonic processes, but by the remains of migrating whales descending to the seafloor. Each carcass became a temporary refuge. Together, countless carcasses formed a network. The scale of time involved deepens the story further. Large whales have been inhabiting Earth's oceans for approximately 35 million years. Throughout that vast interval, generation after generation completed migrations, lived their lives, and eventually sank into the depths. Long before humans possessed ships capable of reaching the deep ocean floor, long before hydrothermal vents were known to science, these events were already shaping patterns of dispersal and evolution. Nothing about the whales required deliberate intention. They were not participants in an ecological design. They simply followed their own existence. Yet every descent delivered nutrients, habitat, and opportunity to environments far below. The deep ocean responded by incorporating those gifts into something larger. Over millions of years, isolated communities became connected. Genetic material moved between distant populations. Species expanded beyond the limits that geography alone might have imposed. Ecosystems, separated by thousands of kilometers, which is about 621 miles, found indirect links through a succession of temporary worlds assembled from bone, chemistry, and time. What appears, from the surface, as a simple ending became part of a much broader continuity. A whale's final journey did not merely sustain life at the place where it settled. It helped weave together distant communities across the seafloor, creating pathways through darkness that remained invisible until science learned how to recognize them. In that sense, the deep ocean transformed mortality itself into connection, building bridges from events that seemed, at first glance, to be nothing more than an ending. Scattered across the deepest seafloor are shapes that seem, at first encounter, entirely geological. Some resemble delicate branches spreading across the sediment. Others look like weathered mineral deposits, fragments of coral, or small structures assembled by forces of chemistry rather than biology. They rest motionless upon the bottom, blending so completely into their surroundings that early observers often struggled to determine whether they were alive at all. Yet, these strange forms belong to living organisms. More remarkably, each one is a single cell. Among the many extraordinary inhabitants of the hadal realm, xenophyophores occupy a category almost entirely their own. They are the largest individual cells known on Earth. The largest recorded specimens reach approximately 20 centimeters across, which is about 0.12 miles. At that scale, a xenophyophore approaches the size of a dinner plate. There are no tissues, no organs, no multicellular body plan. Everything exists within the biological boundaries of a single cell. The achievement becomes even more remarkable when viewed in terms of complexity. A typical cell is measured in microscopic dimensions. It performs its functions within a space invisible to the unaided eye. Xenophyophores have expanded that concept to a scale that appears almost incompatible with ordinary expectations of cellular life. Their bodies accomplish this partly through the construction of elaborate external structures known as tests. These housings are assembled from materials gathered directly from the surrounding seafloor. Sediment grains, mineral particles, fragments of biological debris, and other available materials are collected and incorporated into carefully organized forms. The resulting structures can appear surprisingly intricate, reflecting patterns that seem more architectural than cellular. Certain species demonstrate an especially selective approach. One known xenophyophore constructs its test almost entirely from the needle-like skeletal elements of glass sponges. These tiny fragments are chosen according to size and arrangement, then assembled into a coherent structure through processes directed by a single cell. The scale of the finished construction makes the accomplishment difficult to appreciate. What appears to be the work of a small colony or multicellular organism is instead guided by one immense cellular entity interacting with its environment. The test serves purposes beyond simple protection. Its cavities, surfaces, and internal spaces become habitats for numerous smaller organisms. Bacteria colonize its structure. Tiny worms move through its passages. Microscopic invertebrates establish themselves among the accumulated materials. A miniature ecosystem develops within and around a framework built entirely through the biological activity of a single cell. In this way, xenophyophores become ecological engineers of the deep sea floor. Their presence alters the local environment, creating opportunities for other forms of life to establish themselves where fewer resources might otherwise exist. Their evolutionary history stretches deep into time. Xenophyophores belong to a broader group known as the foraminifera, organisms with a fossil record extending back more than 500 million years. Through countless geological eras, members of this lineage have persisted, while continents shifted, oceans opened and closed, and entire ecosystems emerged and vanished. The materials incorporated into their tests sometimes contain organic particles that descended from surface waters hundreds or even thousands of years earlier. Layer by layer, these structures accumulate fragments of biological history. Embedded within them are records of events that occurred long before the cell itself began growing. As a result, xenophyophores contribute to some of the oldest preserved organic archives available on the hadal seafloor. Their unusual appearance led to decades of scientific uncertainty. Researchers initially classified them as sponges. Later, they were assigned to different groups of simple organisms. At one point, their characteristics appeared so distinctive that some scientists considered placing them within an entirely separate major branch of life. More than a century passed before their position was understood with confidence. The confusion itself reveals something important. Xenophyophores challenge assumptions about what a cell can be. They blur distinctions between individual and community, between microscopic and macroscopic life, between organism and habitat. On the floor of the deepest trenches, a single cell gathers particles from the surrounding world, builds an elaborate home, preserves fragments of ancient history, and provides shelter for an entire neighborhood of smaller creatures. Given enough time, enough isolation, and enough environmental stability, life discovers possibilities that seem almost impossible until they are encountered resting quietly within the sediment, waiting to be recognized for what they truly are. Far beneath the realms occupied by fish, squid, worms, and every other creature large enough to notice, another domain extends through sediment, stone, and fractured crust. It is a biosphere without movement in the familiar sense, without eyes, limbs, shells, or teeth. Its inhabitants leave no tracks and build no visible structures. Yet in terms of abundance, persistence, and sheer influence upon the planet, they may represent one of the greatest living systems Earth has ever produced. The deepest expressions of life belong not to animals, but to microbes. Within hydrothermal vent systems, among fluids emerging from Earth's interior, certain microorganisms occupy conditions that seem almost detached from ordinary biology. One of these is an archaeon known as Pyrococcus furiosus. Its preferred environment reaches 100 degrees Celsius, or 212 degrees Fahrenheit. Water at this temperature would destroy the proteins of most familiar organisms. Cells adapted to temperate oceans would rapidly fail. Yet for this archaeon, such conditions are not extreme. They are ideal. Its enzymes, membranes, and internal chemistry are built around temperatures that surface organisms experience only as catastrophe. When conditions cool below 70 degrees Celsius or 158 degrees Fahrenheit, its growth begins to decline. It does not merely tolerate boiling water, it depends upon it. The environment that appears destructive from a human perspective serves as the stable center of its existence. Elsewhere, beneath the sea floor, life proceeds according to a rhythm so slow that it almost disappears into geological time. Core samples, extracted from depths reaching 86 meters beneath marine sediments, have revealed living microbial communities isolated from surface influences for immense spans of history. These cells are not dormant. They continue the processes associated with life. They consume energy, repair damage, and reproduce. Yet reproduction unfolds at astonishing speeds, or rather, astonishing absences of speed. Some estimates suggest that individual microbial cells in these environments may divide only once every 10,000 years. Entire human civilizations could emerge, transform, and vanish during the interval between two cellular divisions. Continents would shift by measurable distances. Species would appear and disappear. Through all of it, these microorganisms would continue their almost imperceptible existence within the darkness below. The energy sustaining them often originates from sources entirely detached from sunlight and biological productivity at the surface. In certain regions, seawater penetrates deep into fractured rock and reacts with minerals in a process known as serpentinization. This geological interaction releases hydrogen gas. Microorganisms harvest that hydrogen as an energy source, oxidizing it through metabolic pathways refined across immense stretches of evolutionary time. In such systems, life is powered directly by chemistry generated within the planet itself. No plant stands at the beginning of the food chain. No photosynthetic organism captures sunlight. No organic debris drifts downward from distant waters above. The primary energy source emerges from interactions between rock and water. Geology becomes nutrition. The implications gradually expand outward. If microorganisms can persist beneath the seafloor, fueled by chemical reactions occurring within stone, then many assumptions about where life belongs begin to loosen. Surface ecosystems, vast as they seem, become only one expression of a broader phenomenon. Researchers estimate that the total biomass contained within deep marine sediments may rival the combined biomass of all life existing above the surface. Hidden beneath oceans, extending through layers of mud and rock, an enormous living realm carries on beyond ordinary observation. For the organisms inhabiting that realm, the surface would not appear welcoming. Oxygen-rich air, fluctuating temperatures, intense radiation from sunlight, and rapidly changing environmental conditions would represent profound challenges. The habitats humans consider normal would be hostile territory. This perspective introduces a subtle reversal. Life is often imagined as something that originated in illuminated environments and later expanded into deeper settings. Yet, many microbial lineages seem perfectly at home in darkness, pressure, and chemical energy. Their histories stretch backward toward conditions resembling those found in the ancient Earth. From that vantage point, the deep ocean ceases to look like an exception. It begins to resemble a foundation. The familiar world of sunlight and waves becomes only a thin layer resting above a far larger living system, one whose inhabitants have spent countless millions of years thriving beyond the reach of day. Sustained by the planet itself, there is a peculiar contrast hidden within modern exploration. Human beings have walked upon another world, mapped distant planets, and sent robotic spacecraft beyond the outer boundaries of the solar system. Yet much of the seafloor of our own planet remains unseen. Beneath the oceans lies a landscape larger than all of Earth's continents combined, and most of it exists beyond direct observation. Despite centuries of navigation and decades of technological progress, less than 1/4 of the ocean floor has been mapped at the level of detail required to reveal features smaller than a house. Vast regions remain represented only by broad outlines and incomplete measurements. Mountains, canyons, ridges, volcanic fields, and trenches occupy these areas, but many of their details are still hidden beneath kilometers of water. In an unexpected inversion of perspective, the surface of Mars has been mapped with greater resolution than much of Earth's own seafloor. The red planet, separated from us by millions of kilometers, which is about millions of miles, has in many respects become more familiar than the deepest regions beneath our oceans. The consequence of this incomplete knowledge appears every time researchers descend into previously unexplored waters. New species continue to emerge with remarkable consistency. This is not an occasional event celebrated once every few years. It has become an expected outcome of exploration itself. Expeditions entering unsampled environments routinely encounter organisms unknown to science, creatures that have spent countless generations existing beyond human awareness. A striking example came during the Ningaloo Canyons expedition of 2023. Over a span of just 35 days, researchers documented more than 100 species previously undescribed by science. One voyage, lasting little more than a month, expanded the known inventory of life by an extraordinary amount. What makes this particularly significant is that the pace of discovery has not begun to decline. In many scientific fields, progress gradually slows as the most accessible discoveries are made and remaining mysteries become increasingly rare. Deep ocean biology has followed a different path. Advances in technology continue to reveal entirely new territories. Improvements in remotely operated vehicles, imaging systems, sampling equipment, and pressure-resistant engineering have expanded the depths that researchers can reach. Each improvement opens access to environments that were previously inaccessible. As a result, the catalog of deep-sea life does not resemble a library nearing completion. It resembles the earliest shelves of a collection that has only recently begun to form. The most remote environments remain the least explored. Hadal trenches represent perhaps the clearest example, reaching depths between 8,000 kilometers, which is about 4,971 miles, and 11,000 kilometers, which is about 6,835 miles, would be impossible if interpreted literally. So these depths are more accurately understood as 8,000 meters and 11,000 meters within the ocean. Operating scientific equipment under such immense pressure demands some of the most sophisticated engineering ever developed for marine research. Only a small number of institutions possess the capability to conduct such missions. Even when the technology exists, opportunities remain limited by cost, logistics, weather, and the immense complexity of deep ocean operations. There are 37 recognized hadal trenches across the world. Most have been visited only briefly. Some have been examined once and never revisited. Others remain largely unexplored, known primarily through remote measurements rather than direct observation. This context reshapes everything that has come before. Every adaptation described, every unusual body plan, every ecosystem powered by chemistry rather than sunlight, every organism inhabiting depths once considered uninhabitable has been discovered within the fraction of the deep ocean that humanity has managed to examine directly. Beyond that fraction stretches an expanse still waiting to be understood. Most of the organisms living in the darkest parts of the ocean have never been observed by human eyes. Many have never been collected, photographed, or named. Entire branches of ecological relationships may exist beyond current knowledge. Biological strategies unlike any yet described may persist within isolated valleys, trenches, and seamounts hidden beneath the water. The deep ocean remains one of the last places on Earth where discovery is not an exception but an expectation, where every descent carries the possibility of encountering something entirely unfamiliar, and where the unknown still occupies far more space than the known. Far from Earth, beyond the familiar paths of weather, oceans, and continents, several worlds drift through darkness, carrying a possibility that would have seemed far less plausible only a few decades ago. Their surfaces appear frozen and lifeless. Vast shells of ice conceal everything beneath. From a distance, they resemble inactive spheres of rock and frost, suspended in the cold reaches of the solar system. Yet hidden below those frozen exteriors are oceans. Not oceans exposed to sunlight, wind, or sky, but oceans sealed beneath layers of ice thousands of meters thick. In 2005, observations from the Cassini spacecraft confirmed that Saturn's moon Enceladus possesses a liquid water ocean beneath its icy crust. Even more remarkable, that concealed ocean communicates with space through fractures in the ice. Water vapor erupts outward in towering plumes, carrying organic compounds, molecular hydrogen, and other materials from the hidden sea below. The discovery transformed Enceladus from an icy moon into something far more intriguing. It became a place where water, chemistry, and internal energy coexist beneath a protective shell, isolated from the vacuum of space. Elsewhere, orbiting Jupiter, another ocean world waits beneath an even thicker covering of ice. Europa is believed to contain a subsurface ocean holding roughly twice the volume of all Earth's oceans combined. This immense body of water exists in perpetual darkness. No sunrise penetrates its depths. No direct connection links it to the warmth of a nearby atmosphere. Instead, its energy comes from an entirely different source. As Europa moves through Jupiter's immense gravitational field, the Moon experiences continual stretching and compression. This process, known as tidal flexing, generates heat within its interior. The result is a hidden ocean maintained in liquid form, despite the intense cold of the outer solar system. Viewed from a biological perspective, these environments possess a surprising familiarity. Cold water, high pressure, darkness, chemical energy. The combination echoes conditions found around Earth's hydrothermal vent systems. Although separated by unimaginable distances, these worlds share several of the fundamental ingredients present in some of the most productive ecosystems discovered in the deep ocean. This resemblance carries profound significance because of a scientific assumption that once appeared unshakable. Before the discoveries of the late 1970s, sunlight occupied a privileged position in discussions of habitability. Nearly every known ecosystem ultimately depended upon photosynthesis. Plants, algae, and photosynthetic microorganisms formed the energetic foundation supporting larger food webs. Remove sunlight, and the chain appeared broken. The hydrothermal vents changed that understanding. Researchers discovered complete ecosystems operating entirely on chemical energy. Microorganisms transformed compounds emerging from Earth's interior into biological fuel. Entire communities flourished without any connection to sunlight. Energy still existed, but its source came from chemistry rather than illumination. Once that realization emerged, the absence of sunlight could no longer serve as an automatic disqualification for life. The question itself changed. Instead of asking whether a distant environment resembles the sunlit surface of Earth, scientists began asking whether it possesses liquid water, chemical energy, and conditions capable of supporting self-sustaining biological processes. The focus shifted from familiarity to possibility. As a result, many of the organisms now studied as models for potential extraterrestrial life are not animals from forests, grasslands, or coral reefs. They are deep-sea extremophiles. Chemosynthetic microorganisms thriving in darkness have become some of the most important guides in the search for life beyond Earth. Their existence demonstrates that biology can remain coherent under circumstances once considered impossible. They reveal a flexibility within life that continues to expand the boundaries of scientific imagination. The trenches and vent systems of Earth's oceans have therefore become more than destinations for marine exploration. They function as natural laboratories for astrobiology. Every adaptation discovered there, every metabolism powered by unusual chemistry, every organism surviving beyond previous expectations contributes to a broader effort to understand where life may emerge and persist. In this way, the deep ocean reaches far beyond the planet itself. The discoveries made in its darkness extend outward toward distant moons and hidden seas. By revealing how life operates under conditions once thought uninhabitable, the deep ocean has enlarged the scope of one of humanity's oldest inquiries, transforming it from a question about Earth into a question about the wider universe. While continents shifted, mountains rose and eroded away, ice sheets advanced and retreated across the surface of the planet. A different world endured beneath the oceans with remarkable consistency. The deep sea floor exists within one of the most stable environments Earth has ever produced. Across approximately 50 million years, temperatures in the deepest regions of the ocean have changed by less than 2 degrees Celsius, or 36 degrees Fahrenheit. Chemical conditions have remained comparatively steady across spans of time that dwarf human history. Darkness has persisted uninterrupted since long before complex animals first diversified across the planet's oceans. The cycles that dominate life at the surface, seasons, droughts, glaciations, storms, and shifting climates, arrive only as faint echoes in the depths. This stability has carried profound consequences for the history of life. Throughout Earth's past, the surface world experienced repeated biological crises. Entire ecosystems vanished. Vast numbers of species disappeared in relatively brief intervals. The Ordovician extinction, the Devonian extinction, the Permian extinction, the Triassic extinction, and the Cretaceous extinction each reshaped the living world. Some eliminated more than 3/4 of all known species. Others approached the destruction of nearly all complex life then in existence. Yet the deepest ocean often experienced these catastrophes differently. Conditions there could certainly be disrupted. Food supplies might decline. Ecological relationships could shift. Some species disappeared. But the fundamental characteristics of the environment remained comparatively buffered from the most extreme changes occurring above. The darkness did not vanish. The pressure remained. The surrounding water continued to provide a degree of chemical and thermal stability unavailable elsewhere. As a result, lineages that could not survive at the surface sometimes endured below. The extinction event that occurred approximately 66 million years ago provides one of the clearest examples. This catastrophe ended the age of the non-avian dinosaurs and eliminated more than 3/4 of Earth's species. Forests collapsed, food webs unraveled, entire groups disappeared forever. Deep ocean communities were affected, yet many were not annihilated. Life persisted within habitats insulated from some of the immediate consequences unfolding above. In places where surface ecosystems experienced abrupt transformation, the depths provided continuity. Evolutionary branches severed elsewhere retained living representatives below. Certain organisms embody that continuity with extraordinary clarity. The nautilus belongs to a lineage extending back more than 500 million years. Ancestors of these animals moved through ancient seas long before the first dinosaurs appeared. They survived every major extinction event known from the fossil record. Their persistence is not accidental. Nautiluses inhabit deeper waters, where environmental conditions remain relatively stable. Depth became a form of shelter, preserving a biological design through eras that erased countless others. Even individual organisms can span astonishing stretches of time. Some deep water sponges are estimated to exceed 10,000 years of age. Such creatures were already alive before agriculture emerged in many regions of the world. They existed before cities, before written language, before recorded history. Year after year, century after century, they filtered water and grew at almost imperceptible rates, while entire civilizations rose and disappeared far above. These organisms are more than survivors. They are living connections to environments and evolutionary experiments that would otherwise be known only through stone. The deep ocean contains body plans, physiological strategies, and ancient lineages whose relatives vanished from shallower habitats millions of years ago. Some represent solutions to survival that originated in geological eras fundamentally different from the modern world. Others preserve branches of evolutionary history that escaped repeated waves of extinction simply because they occupied places less vulnerable to planetary upheaval. For this reason, the deep ocean can be understood as something more than unexplored territory. It functions as an archive, not an archive of paper, carved tablets, or stored records, but one written in living organisms. Within its communities remain traces of ancient biological worlds, preserved through continuity rather than documentation. Species carry histories within their anatomy, behavior, and genetic inheritance. Entire ecosystems preserve evidence of pathways evolution followed long ago. Far below the reach of sunlight, Earth has maintained a record of itself across immense stretches of time. While the surface repeatedly reinvented its living world, the deep ocean retained fragments of what came before, holding them in reserve within a realm where change moves more slowly and memory can endure for millions of years. Imagine a chain of rooms scattered across a vast landscape, each sealed from the others for immense spans of time. The temperature remains nearly constant. The rules governing survival remain largely the same. The inhabitants begin with shared ancestry, carrying similar biological inheritance into each isolated space. Then the doors close, and evolution continues separately within each room. Something very similar occurs in the deepest trenches of the ocean. Although hadal trenches belong to the same planet and share many environmental characteristics, they are often separated by enormous stretches of abyssal seafloor. Between them lies terrain that functions as a formidable barrier for organisms specialized for trench life. Populations that once shared common ancestry become isolated, unable to exchange genetic material freely. Over time, those populations begin to diverge. The process unfolds gradually, generation after generation, far beyond the span of direct human observation, yet its results accumulate with remarkable consistency. One of the clearest examples involves hadal snailfish. Populations inhabiting the Mariana Trench and the Yap Trench descended from a common ancestor, yet genomic analyses indicate that their evolutionary paths separated approximately 920,000 years ago. In geological terms, such a period represents a relatively recent event. Continents remain essentially unchanged. Ocean basins preserve familiar outlines. For evolution, however, 920,000 years provides ample opportunity for divergence. The descendants occupying each trench still resemble one another. They remain recognizably part of the same broader lineage, yet detailed examination reveals differences beginning to emerge throughout their biology. Variations appear in the concentration of pressure protecting molecules. Differences arise in gene copy numbers associated with survival under extreme conditions. Sensory systems begin to reflect the particular demands of each environment. The changes are subtle when viewed individually, but together they form the early architecture of separation. What makes these discoveries especially powerful is the method through which they are revealed. Earlier generations of scientists often depended upon fossils to reconstruct evolutionary history. Bones, shells, and preserved impressions served as fragments of evidence from vanished worlds. Today, researchers can examine the genomes of living hadal organisms directly. Within those genomes lies a record of ancestry. Mutations accumulate over time. Genes duplicate, disappear, or change function. Patterns of similarity and difference reveal relationships, extending backward through countless generations. Scientists no longer need to infer every aspect of divergence indirectly. In many cases, they can read portions of the evolutionary story directly from the organisms themselves. Each trench, therefore, becomes more than a habitat. It becomes an ongoing experiment. Pressure remains immense. Food remains scarce. Temperatures remain cold. Darkness persists without interruption. The broad environmental constraints are similar, yet each trench possesses unique geography, chemistry, ecological interactions, and historical circumstances. As a result, evolution explores slightly different pathways in each location. The question unfolding within these isolated environments is both simple and profound. What happens when a vertebrate lineage experiences the same fundamental pressures but follows its own independent trajectory for hundreds of thousands or millions of years? Different trenches produce different answers. Some lineages emphasize one adaptation, others refine another. Genetic solutions emerge that are shared, while others remain unique to particular populations. By comparing these outcomes, biologists gain insight into the balance between predictability and variation in evolution itself. The deep ocean offers a perspective rarely available elsewhere. Surface ecosystems are interconnected through migration, climate fluctuations, and continual ecological exchange. Hadal trenches provide a contrasting situation, places where separation can persist long enough for evolutionary processes to become especially visible. In that sense, the deepest trenches function as natural laboratories operating across immense stretches of time. No researcher designed them. No institution maintains them. Yet, they continue their work with extraordinary patience. While human lives pass in decades, these environments conduct experiments measured in hundreds of thousands of years. Every generation leaves a small genetic mark. Every isolated population drifts a little further from its relatives. Over time, those differences accumulate into something entirely new. Far below the reach of sunlight, evolution continues its patient work, recording outcomes not in notebooks or databases, but within the genomes of living organisms, each trench preserving its own version of a story still being written. A map appears chaotic only when its symbols are unfamiliar. Seen from above, the deep ocean often seems filled with contradictions. Bodies fuse together. Skeletons soften instead of strengthening. Eyes disappear. Stomachs vanish. Skin becomes darker than coal or nearly transparent. Light emerges from living tissue in a place where sunlight never arrives. The first impression is one of disorder, as though evolution abandoned its usual rules and began experimenting without restraint. Yet the deeper one looks, the more a different pattern emerges. Beneath every unusual structure lies a chain of cause and effect, extending back through countless generations. Each adaptation is connected to a problem. Each problem emerges from the environment. Follow the chain carefully enough, and what once appeared strange becomes inevitable. Consider the anglerfish. In the vast darkness of the deep ocean, finding another member of the same species can be extraordinarily difficult. Distances are immense. Populations are sparse. Encounters may occur only rarely during an individual's lifetime. Under such conditions, a male that permanently attaches to a female and becomes part of her body solves a fundamental problem. The arrangement appears extraordinary from a surface perspective, yet within that environment, it represents a direct answer to the challenge of reproduction. The same logic extends to the giant tube worm. Most animals acquire nutrition through mouths, digestive systems, and feeding behaviors refined across evolutionary history. Hydrothermal vent ecosystems present a different opportunity. Chemosynthetic microorganisms already possess the biochemical machinery required to transform chemical compounds into usable energy. Hosting those microorganisms directly within living tissue removes the need for a conventional digestive system. The worm does not abandon digestion because it failed to evolve one. It abandons digestion because a more effective solution became available. The Mariana snailfish reveals another layer of the same principle. To a surface observer, its partially ossified skeleton and incompletely closed skull can appear unfinished. Yet rigid structures face particular challenges under immense pressure. Flexibility distributes force more evenly than resistance. Bone that remains somewhat compliant avoids stresses that a more heavily reinforced design might experience. What seems fragile from one perspective becomes remarkably well suited from another. Even coloration follows the same logic. In regions where bioluminescence provides the primary source of illumination, visibility becomes a matter of survival. A single reflected photon may reveal the presence of predator or prey. Ultrablack skin absorbs extraordinary amounts of light because every fraction of additional absorption carries potential advantage. The difference between reflecting a small amount of light and reflecting almost none at all may determine whether an animal remains concealed. The recurring appearance of bioluminescence offers perhaps the clearest demonstration. This ability evolved independently dozens of times. Different organisms arrived at similar outcomes through separate biochemical pathways. Such repeated convergence suggests a powerful underlying pressure. In a world dominated by darkness, producing light becomes useful for communication, hunting, defense, camouflage, and reproduction. The pathway may vary, but the destination remains similar because the environment repeatedly rewards the same capability. Viewed together, these examples reveal a consistent theme. None of these organisms represent evolutionary accidents. None are biological mistakes. Each embodies a successful response to a specific set of constraints. Pressure, cold, darkness, scarcity, isolation, and chemistry act as filters, removing ineffective designs and preserving those capable of persistence. The deep ocean, therefore, challenges a subtle assumption carried by surface dwellers. Human intuition develops within environments shaped by sunlight, abundant oxygen, changing weather, and familiar ecological relationships. Those conditions become the standard against which other habitats are judged. Yet the hadal world was never obligated to follow that standard. Its rules emerged from different physical realities. Its challenges demanded different solutions. The creatures inhabiting it evolved according to those realities, with extraordinary precision. What appears improbable when measured against surface expectations becomes entirely coherent when understood within the context that produced it. The apparent madness dissolves into structure. The unfamiliar becomes understandable. Beneath every adaptation lies a logic as rigorous as mathematics, written not in equations, but in living bodies, shaped across immense stretches of evolutionary time, each one carrying the record of a problem solved exactly as the environment required. Patterns become most visible when they emerge repeatedly from places that cannot communicate with one another. Across the deep ocean, trenches lie separated by immense distances. Some are divided by thousands of kilometers, which is about 621 miles for every 1,000 kilometers crossed of abyssal terrain. Their inhabitants often share no recent connection. Their histories unfold independently. Millions of years pass with little opportunity for exchange. Yet when biologists compare the outcomes, they encounter a remarkable result. Again, and again, evolution arrives at similar solutions. Different lineages confront the same physical realities and produce familiar answers. Pressure-resistant chemistry appears repeatedly. Specialized mechanisms for operating in darkness emerge over and over. Bodies become softer, more flexible, and more energy efficient. Strategies for detecting food, conserving resources, and navigating scarcity converge despite originating from unrelated ancestors. The repetition is difficult to ignore. Convergence is a well-known feature of evolution. Wings evolved separately in insects, birds, and bats. Streamlined shapes appear in different aquatic animals. Similar ecological pressures often guide unrelated organisms toward comparable designs. The hadal realm extends that principle into one of its most striking forms. The similarities occur across isolated trenches, distant lineages, and enormous spans of time. They appear not as occasional coincidences, but as recurring outcomes. The same environmental challenges repeatedly shape life toward the same general destinations. This observation suggests something profound about where those destinations originate. It is tempting to imagine that every adaptation exists as a hidden possibility waiting inside a genome, ready to emerge when circumstances permit. Yet the patterns observed in the deepest ocean point attention elsewhere. The common factor is not ancestry, it is physics. Pressure influences the architecture of proteins. Cold affects the speed and efficiency of enzymes. Darkness changes the value of vision and the usefulness of light production. Scarcity rewards conservation and punishes waste. These are not biological preferences. They are properties of the environment itself. Long before any organism evolved to confront them, the underlying rules already existed. Life does not create those rules. Life responds to them. In that sense, many extraordinary deep-sea adaptations can be understood not as inventions, but as discoveries. The chemical compounds that stabilize cellular machinery under immense pressure were always permitted by the laws of chemistry. The biochemical pathways, enabling chemosynthesis, were always available within the possibilities of matter and energy. Ultrablack surfaces, capable of absorbing nearly all incoming light, were always compatible with the physics of photons. Counter-illumination, where an organism produces light to conceal itself, emerges naturally from the interaction between vision and darkness. The environment presents a problem. Physics restricts the range of viable answers. Evolution explores that range. Given sufficient time, successful solutions emerge. This perspective transforms the deep ocean from a collection of biological curiosities into something much larger. It becomes a demonstration that certain outcomes may be inevitable whenever particular conditions persist long enough. Cold water, high pressure, permanent darkness, limited energy. These circumstances repeatedly guide evolution toward similar destinations because the underlying constraints remain the same. The organisms differ, the history differs, the route differs, yet the destination often resembles those reached elsewhere. As this realization settles into view, the significance extends beyond Earth itself. Earlier generations often imagined life elsewhere by searching for environments resembling familiar landscapes at the surface of our planet. Forests, rivers, sunlight, and temperate climates seemed essential because they were familiar. The deep ocean altered that assumption. It revealed that biology can flourish under conditions once considered hostile beyond endurance. If the recurring solutions of the hadal world arise from the physics of cold, dark, pressurized water, then those solutions are not necessarily tied to a particular species, continent, or evolutionary history. They belong to the broader rules governing matter and life. Far beneath the waves, the deepest ocean offers a subtle lesson. The patterns discovered, there may not be local exceptions. They may be expressions of principles far older and more universal than any single world, principles written into the fabric of nature itself, waiting wherever similar conditions endure long enough for life to find them. There are places on Earth where an entire human lifetime can pass without leaving the faintest trace upon the surrounding environment. The deepest regions of the ocean belong to that category. At approximately 8,000 meters below the surface, which is about 4,971 miles for every 8,000 kilometers if measured horizontally, although depth itself descends vertically into the water column, the familiar soundtrack of the world has long since faded away. The atmosphere that shapes life above has no meaningful influence there. Storms can rage across oceans. Hurricanes can churn the surface into mountains of water. Rain can fall for days. None of it reaches these depths in any recognizable form. What remains is a realm defined by extraordinary stillness. The sounds familiar to terrestrial life originate from constant movement. Wind passes through forests. Waves collapse against shorelines. Rivers tumble over stone. Countless animals call, hunt, communicate, and compete. Human civilization contributes its own layer of activity: engines, machinery, transportation, and industry. Together, they create an acoustic environment of continual change. Far below, much of that complexity disappears. The water column absorbs and diminishes disturbances descending from above. By the time one reaches the hadal zone, only rare sounds interrupt the surrounding calm. A crustacean may produce a sharp click. Geological forces deep within Earth's crust may release a distant groan as tectonic plates shift imperceptibly against one another. Hydrothermal activity may generate a faint chemical hiss where heated fluids meet colder seawater. Between such events stretch intervals of remarkable stability, yet sound is only part of the story. The darkness of the deep ocean differs fundamentally from the darkness experienced at the surface. A room at night remains connected to the wider universe. Starlight enters through windows. Moonlight reflects from clouds. Even beneath overcast skies, scattered photons persist. The deepest trenches possess no such connection. No photon from a distant star has ever completed the journey through kilometers of seawater to reach the floor of the Mariana Trench. The darkness there is complete. It does not arrive with sunset and depart with dawn. It remains constant across days, centuries, and millennia. It is a darkness older than forests, older than animals walking on land, older than the oxygen-rich world familiar today. Within such conditions, life experiences time differently. On the surface, rapid cycles dominate existence. Days alternate with nights. Seasons return each year. Plants grow, flower, and fade. Generations succeed one another in quick succession. In the deep ocean, some biological clocks proceed at a vastly slower pace. Certain microorganisms divide so infrequently that a single reproductive event may occur once every 10,000 years. During one cellular division, entire chapters of human history could unfold and vanish. The organism remains active, metabolizing, persisting, and participating in its environment, yet at a tempo almost indistinguishable from geology itself. Other inhabitants embody similar scales of duration. Deep water sponges may remain alive for 10,000 years or more. Individual organisms can persist through spans of time, reaching back before the first cities appeared. Their growth is measured not in seasons, but in epochs. Even evolutionary lineages reveal this altered relationship with time. The Mariana snailfish lineage has occupied the trench environment for approximately 20 million years. Throughout climatic upheavals, shifting coastlines, advancing glaciers, retreating glaciers, and repeated transformations of surface ecosystems, life continued in the depths according to its own rhythms. Events that reshaped continents passed overhead without fundamentally altering the darkness below. The perspective created by such continuity is difficult to grasp. Human civilization often treats permanence as something measured across centuries. Ancient monuments seem enduring. Nations appear stable. Languages feel established. Yet, against the backdrop of the deep ocean, these durations become brief intervals. The trenches existed before our species emerged. They existed before the earliest members of our genus appeared. They existed before many familiar ecosystems took their modern form. Their defining conditions have persisted across spans of time so vast that much of human history occupies only the thinnest fraction of the record. Far below the surface, where darkness remains unbroken and change unfolds with extraordinary patience, permanence acquires a different meaning. Things that seem ancient above become recent arrivals, and much of what humanity considers enduring has only just begun. Long before the first story was told beside a fire, before the first city rose from stone, before the earliest ancestor recognizable as human walked across a landscape beneath the sun, life was already continuing its work in the deepest parts of the ocean. It has never stopped. The entire span of recorded human history occupies only a brief interval when measured against the age of deep ocean ecosystems. Every empire, every migration, every language, every scientific breakthrough, every conflict, every monument, and every work of art belongs to roughly the last 10,000 years. By comparison, many deep ocean lineages trace their ancestry across hundreds of millions of years. Those two timelines overlap only at the very end. From a human perspective, our species occupies the center of experience. History is organized around human events. Maps emphasize human settlements. Time itself is often divided according to human milestones. Yet, far below the surface, none of those milestones possess any significance. A Mariana snailfish moving through the trench encounters its world through pressure, chemistry, scarcity, and opportunity. When a research vehicle descends carrying cameras and instruments, the fish does not recognize a technological achievement. It does not perceive exploration, discovery, or science. If it approaches a baited trap, it does so for the same reason its ancestors approached food sources long before human beings existed. Food remains valuable. Energy remains scarce. The logic governing its behavior has not changed. The same perspective applies to hydrothermal vent ecosystems. When researchers first encountered these communities during the late 20th century, the discovery transformed biology. Entire ecosystems existed independent of sunlight. Organisms flourished through chemical energy pathways once thought insufficient to support complex communities. Textbooks required revision. Assumptions were reconsidered. Yet nothing changed within the vents themselves. The tube worms continued growing. The microorganisms continued processing chemical compounds. The surrounding animals continued feeding, reproducing, and competing. The ecosystem had been functioning continuously for immense spans of time before any human observer arrived. It was not awaiting recognition. It did not become more real when it entered scientific literature. Its existence never depended upon being seen. This quality of indifference distinguishes the deep ocean from many environments altered extensively by human activity. Across much of the planet, biological communities now exist within landscapes reshaped by agriculture, industry, transportation, and urban development. Human influence reaches forests, rivers, grasslands, coastlines, and even the atmosphere itself. Few places remain entirely separated from those effects. The deepest trenches represent one of the rare exceptions. Their pressure, depth, and remoteness have created barriers difficult to cross even with modern technology. As a result, many of their fundamental biological processes continue much as they did before human civilization emerged. Evolution proceeds. Ecosystems function. Life adapts according to pressures that originate from the environment rather than from human activity. This indifference should not be mistaken for hostility. The deep ocean is not resisting humanity. It is not opposing exploration or rejecting observation. Opposition requires relationship. The deepest ecosystems possess almost none. They simply continue. While cities expand, governments change, technologies advance, and cultures transform, organisms thousands of meters below remain engaged in entirely different concerns. They search for food. They reproduce. They survive. Generation follows generation according to rhythms established long before human history began. There is a certain clarity in recognizing this. At this very moment, life persists in places where no unprotected human body could survive. Creatures move through darkness that has remained unbroken for spans of time, exceeding the existence of our species. Microorganisms continue chemical processes older than civilization. Entire communities operate without awareness of the world above. The realization gently alters perspective. The deep ocean was not designed with humanity in mind. It was not constructed to inspire, challenge, or accommodate us. Its existence follows no human narrative. It simply occupies its place within the planet, vast and enduring, carrying on with extraordinary complexity, whether observed or not. For hundreds of millions of years, it has been shaping life according to its own rules. Only recently did we begin to notice. Even now, the greater part of that story continues beyond our sight, unfolding exactly as it always has. At this moment, while cities glow beneath evening skies and daylight reaches coastlines half a world away, life continues in a place almost completely disconnected from the rhythms that define human experience. Far below the western Pacific Ocean, at a depth of approximately 8,000 meters, which is about 4,971 miles for every 8,000 kilometers measured horizontally, a small translucent fish moves through water only 2 degrees Celsius or 36 degrees Fahrenheit. Around it rests a pressure so immense that human intuition struggles to grasp it. The force pressing upon its body is roughly comparable to the weight of 1,600 elephants concentrated above every exposed surface. Yet the fish moves without strain. Nothing about its motion suggests difficulty. Nothing about its environment suggests crisis. To it, this is simply home. Its appearance seems almost unfinished when judged by the standards of surface animals. Its body lacks heavy armor. Its skin carries little pigment. Scales are absent. Through translucent tissue, internal organs can sometimes be seen. Its skull never completed the rigid enclosure, familiar in many vertebrates. Yet, every feature exists for a reason. The softness of its bones is not a deficiency. The transparency is not incompleteness. The absence of elaborate defensive structures is not failure. Across millions of years, natural selection removed what was unnecessary and preserved what worked. What remains is not a rough draft. It is a final design shaped specifically for one of the most extreme environments on Earth. In that sense, the fish is among the most precisely fitted vertebrates the planet has produced. It feeds in darkness that has remained uninterrupted for spans of time far exceeding the existence of our species. Its ancestors occupied similar depths through climatic upheavals, shifting continents, advancing ice sheets, retreating ice sheets, and transformations of surface ecosystems beyond counting. The world above changed repeatedly. Entire landscapes appeared and vanished. Species rose, flourished, and disappeared. Little of that reached the trench. For approximately 20 million years, some version of this body plan has continued its existence in the depths. Generation followed generation, without reference to events occurring above. Evolution refined details, adjusted physiology, and improved survival, yet the fundamental relationship between organism and environment endured. Recently, human technology reached that world. In the year 2023, researchers lowered cameras into the trench and recorded these animals in their native habitat. For the scientists involved, the achievement represented decades of engineering, planning, and exploration. Specialized equipment crossed a boundary few machines can survive. For the fish, the encounter meant something much simpler. Food appeared. It approached the bait. The interaction ended. That brief exchange serves as a reminder of how different the scales of significance can be. Everything described throughout this journey, every unusual adaptation, every ecosystem fueled by chemistry rather than sunlight, every lineage shaped by pressure, scarcity, and darkness, emerged from the comparatively small portion of the deep ocean that humans have directly examined. Vast regions remain unexplored. Countless organisms have never been observed. Entire ecological relationships likely continue beyond the reach of current knowledge. At this very moment, life is unfolding in those places exactly as it did before the first research vessel crossed an ocean. The deep sea does not require observation to possess complexity. It does not require recognition to possess beauty. Long before human beings developed language, tools, science, or curiosity about the depths, extraordinary biological worlds already existed there. They continue still. Somewhere below the deepest regions, familiar to us, beyond the range of natural human survival, an organism is moving through darkness, untouched by sunlight. It follows instincts refined across immense stretches of time. It occupies a world governed by rules unlike those of the surface, yet perfectly suited to the life it contains. The ocean does not pause for witnesses. It continues. And in that ancient darkness, where pressure, cold, and time have shaped existence according to their own patient logic, something remains alive, exactly adapted to its place, carrying on a story that began long before humanity arrived and never depended upon being seen.